So please be seated all.
Hello.
So today I have this session, 5 presentations up to 10:20. So now it's 9 o'clock, it's about to start. I hope all the
Hello.
presentations are here.
Hello.
And there are some instructions for oral presentation. I'll just read out these presentation instructions. The presentation time will be strictly enforced. Faculty, professors or postdoctoral scientist presentations, total presentation time is 20 minutes, 70 minutes for the presentation and 3 minutes for question answer. For students presentation, total time is 15 minutes. A warning bell will ring 5 minutes before the end of each presentation, followed by a final bell 2 minutes later. The remaining 3 minutes are allocated for question answer session. There will be a student competition for the best oral presentation. The official student presentations are highlighted in the provided list. Fortunately, we don't have any student presentation here for competitions.
All right, good morning, everybody. We have our first presenter today, Art from Warsaw University. The presentation title is Informal Green Spaces on the Frontline Between Particulate Matter Pollution and Urban Residents. I welcome Art, please. It's all yours.
And very exciting day. Also, thank you for very kind invitation. So I will read my name. My name is Arek Przybysz. I'm from Warsaw University of Life Sciences, which is the agriculture university of Warszawa. The topic of my speech is about informal urban greenery as a potential tool to mitigate particulate matter pollution. I will start with some theoretical information why this topic is important for us. So we are a European country, Poland, which suffers from air pollution. Almost all kinds of pollutants, they are in increased concentration, including PM, which is a small particulates in a size of 1 nanometer to 100 micrometers. We do a lot of effort. As you see on the map, the color is changing from red brick to more green to decrease air pollution. Year by year, we have some good results, but unfortunately, we still suffer from pollution, and concentration of pollutants are way higher than in other developed European countries. Just to tell you, 72% of Polish cities still do not reach the standards for air quality, and every year European Union is preparing a list of the most polluted cities in terms of air quality, and out of 50 cities, 36 are from Poland, like year by year. So it's a lot. Why PM is a problem? It's a health issue. Those particulates are small. They can enter your respiration system. They can reach your lungs, and if they are small enough, they can reach also your bloodstream. They are a reason of many deaths annually in the world. In Poland, it's around 50,000, which is more than 10% in Europe, which is really high. What kind of tools we have to decrease pollution level in the atmosphere? So basically, we can do two things. One is to decrease the emission level. So we do, as globally, but also in Poland, we do a lot of effort. But this is first difficult, and secondly, this is also expensive. So we can replace cars, the old ones, with new electric vehicles. We can invest in new technology, in industry. All this is done in Poland. Unfortunately, we are improving, but this is not enough. Then the second possibility is to remove pollutants which are already emitted, and they are polluting in the atmosphere. And this is first even more difficult and even more expensive. Of course, we have some mechanical filters that we can install in the cities. We have some facilities like that in Poland. But first, expensive, and secondly, more important, they work very locally. We have some road surfaces or paints on the building that can absorb pollutants from the atmosphere. But again, this works very locally, and the efficiency of these paints, they are decreasing with the time. So for us, the only solution which is possible in a bigger scale are plants. We are working with this topic since 20 or 25 years, and for us, plant is a perfect filter. It's a kind of compromise. So if you can see the tree on my photo, you can probably see two things. First, its density. So you can see that the plant is dense. And secondly, you can also see a porosity. So the plant is a perfect compromise between being very dense and very porous. The second thing is also that plants can be planted very close to emission sources, and we can plant them almost everywhere. Now our work is to choose the best plants, not even species, but communities, use them as a tools because different plants, they have different applications, and with the plants to reduce air quality. Also, you can see a leaf and the leaf structure, and here every leaf works as a little air biofilter. So leaves, they are not glossy, the structure is complicated. They are covered with waxes, which are lipophilic, they are covered with hairs, and all those structures that can absorb pollutants. What we know now, we know that plants which are grown close to the roads, as you can see here, and parks, they are very efficient with biofiltrating the air. But most of those plants are very aesthetic, they are grown in city centers. But what we don't know, how the air quality can be affected by informal urban greenery. And this is the topic of my study, and here you have explanation why for us is important. So I don't know how it is in your countries, but in Warsaw at least, 70% of our greenery is informal urban greenery. What I understand by informal greenery, those are parts of urban greenery which are not maintained by administration. It can be very different types of plants, of plants community. Can be forests, can be like locations which are taken by plants, they are not maintained, there are no plants for these areas. All of them, they are not treated by humans. And I think this is kind of true for every city in Poland. So what we did in our research, we selected like tens of locations like that, close to the Wisła River, close to industry, close to the main road, almost everywhere. And we also selected several parks to serve us as a control. What we did first, so first we tested, I don't know how many, also tens of species, just to evaluate which of those wild species are the best in air biofiltration. And you can see some results here, just for few species, you can see the clear differences that one species like Robinia pseudoacacia can accumulate a lot of particulate matter, others like Acer negundo less. but below the graph with particulate matter, you can see the table with heavy metals. And what is interesting, the patterns there is a bit different. So Robinia, which accumulates PM, doesn't accumulate well metals. On the other hand, grasses and acer, they are able to accumulate metals. So our first idea is that we have to keep biodiversity in these locations. Of course, our impact to those places is usually very low, but the conclusion is that more diverse those areas are, better they can biofilter the air. If you want to see some summary, so this is from our 2-year study. You can see the average, how much air pollutants, mostly particulate matter, can be accumulated by park, which is located in the city center, compared to informal urban greenery located somewhere in the city. So the difference is significant. How this is possible? I like this photo very much. This is possible because informal urban greenery quite often is located very, very close to emission sources. So here you have one example from Warsaw. You don't see it on the right side of the photo. You should see a cement plant, so a place where they produce cement. Then you have unfortified roads, which is crossed by big trucks quite often, like every 10 minutes. You have plants, and behind the plants, you have new settlements, like new housing area. So you can imagine the area without those plants. When we started to do this research, people living behind those plants, they are just approaching us, asking what we are doing. We are saying to them, we are measuring the quality of the greenery in this location. Their first question was, will you cut that? We said, I don't know why. Let's see. No, it's bad. They look bad. There are some animals, insects. Maybe the drunk people can stay there. We are afraid of that. And surprisingly, when we continue the research after 2 years, they come back again. And the next year, they said to us, so what are your results? And we said, nice, promising. And they said, very good, leave it. Because if not that, all the dust from the road, from the cement plant, would reach their places. Here you have the leaves of Robinia, which are fully covered with particulate matter. We also did some other research just to show how to improve the efficiency of biodiversity. This is a collaboration with Chinese partner, and our result shows that it's very important to keep also the water bodies. So every lake, every stream, river, increase the humidity in the atmosphere, increase also humidity on the leaves of the plants, and that increase significantly the efficiency of plants to biofilter air. This effect is significant up to 300 meters, but you can notice some differences up to 500 meters, which is significant. Another, for our climate, super important thing is winter. So we are now in tropical zone, so you don't know what it means that in your country you have winter. So when you ask me when is the biggest air pollution in Poland, my answer is winter, because we heat our houses. Most people use cars in the winter. In the summer we can bike. While the plants are in a leafless stage during the winter. That's one of the main questions that we have. So what we do, we also suggest to increase the share of evergreen plants, but with those we have a great problems, because evergreen plants, like conifers, they are able to keep leaves or needles longer than one vegetative season. It also means that they accumulate pollutants for more than one season, and the pollution load can be so high that they cannot stand it. They just start to die back. So we are selecting the most tolerant evergreen plants, but we are also looking for alternative solution. And here you have one of the examples how city used our knowledge. This is carpinus plants, and carpinus plants, they are keeping leaves but dead for the whole winter. The new leaves, they are appearing in spring, and then the old leaves are shed. So of course this filter is not that functional and that efficient as a living plant, but it's still better than plant without leaves. So our suggestion is, and the cities in Poland, I have to admit, they follow it, just to increase the share of this kind of plants. Of course, accumulation of pollutants on leaf surface is not without consequences. As you can see here, higher the accumulation of pollutants is, lower the efficiency of photosynthetic apparatus is. So we know that those plants are suffering. We also did some research on that topic, and we know that more stressed plants are, the least efficient they are, and the least PM they can accumulate. If it sounds obvious for you, I have to say that it wasn't obvious for us, because when plants are affected by slight or moderate stress, they have some defense mechanisms that can increase biofiltration effect. For instance, plants, they produce more waxes. Waxes are important to prevent uncontrolled water losing, but at the same time, they are very good in accumulating of lipophilic particulates. But the results are, as you can see, bigger the stress is, the least efficient plant are. So this cannot be used as a suggestions for a city authorities, because they will not improve the condition in location we are working with. But our idea is to plant the most tolerant plants. More tolerant plant is, probably better it will filtrate the air. This is maybe not related to the topic of my presentation, like directly, and also maybe it's a little bit farther from the topic of the conference, but I think that currently doing only like air biofiltration is not enough. What we are now trying to do, our new goal, is to work in a bigger group involving scientists from other fields, and not to work with air pollution only, but to work with ecosystem services. So here you can see a brief summary of what we did with informal urban greenery. I just selected some lines because this table was very long. So you can see the comparison of how much informal and formal urban greenery can sequestrate carbon, how much it can be involved in water retention, in cooling processes, but also aesthetic, cultural values, and biodiversity. This is also something that we present to city authorities, like my city, Warsaw, and fortunately they understand this kind of tables. Now in Warsaw we are trying to make something which is called Warsaw Biodiversity Index, which is used for buying houses. In the past, when you were buying a house, the price that you have to pay was for a size of the flat, location, if it's close to public transport, if it's close to city center. Now the value of plants will be affected also by nature. So do you have greenery close? What kind of greenery it is? Do you have a water body close? So I think that's a big achievement. And then our goal is to present maps like this one. This is one of the districts of Warsaw, new one, which is developing very fast, and from very natural, it starts to be like highly urbanized. And we communicate to city authorities of this district and presenting in data how the changes in greenery affect ecosystem services. So from this graph, those maps, you can see differences from 19th century to today, the reduction in plants, and also ability of plants to filtrate air. A more green color, it means that plants are more efficient in biofiltration. More brick color, it means that there is no biofiltration of air. I know that it's impossible that the city will come back to this situation from 200 years ago, but we want them to keep a greenery which is valuable and it's still there. And another topic, which I have to be fast, yes, also some places that we cannot grow trees because it's safety reasons are close to the roads, close to the cross sections. So those places should be used by meadows and by lawns. Lawns, they have no ecosystem services, so our goal is to work with meadows. So we started with selection of plants. We tested, I think, that more than 100 species of herbaceous plants, grasses, but not only, of course, if they are good in air biofiltration. Based on this, we created a mix that can be planted close to the roads, Of course, not everywhere. This cannot be planted close to the, I don't know, president palace, but on the ways out of the city, this can be a very efficient tool. You can see a comparison in PM accumulation between the lawn and in between the meadow. What is surprising for us, there is no big effect of plant itself, like its characteristic. The thing that really matters is a biomass. So bigger meadow you have, taller, higher plants you have, more efficient they are. And they are also extremely tolerant to drought, to salinity, to city conditions. So you can see the photo here. The lawn is yellow. This is how lawns in Poland look now in the summertime. And back in the photo, you can see a green meadow. So this also shows that those communities, meadows, they can also serve valuable ecosystem services when grasses are not functional. And again, very important for us, meadow can be kept all over the year. I know that in winter it doesn't look nice, but again, this is a tool that you will not use everywhere. We have locations where aesthetics are not that important, but because they are very close to the emission source, which are cars, they can accumulate lots of pollutants. I think that the value for winter is here highly underestimated because we have also a lot of snow, and the snow is taking PM very easily from the plants. So we believe that a lot of pollutants are stabilized also in the soil. So now many places in Warsaw, the meadows are kept all over the winter and they are mowed in March. A new idea is also to work with microplastics. So this is a new data. We also show that these herbaceous communities can accumulate that, and thank you very much.
Now the session is open for discussion.
Hi, Arcadius. I'm very glad of your talk because we are also working on something similar. My question is,
Okay, so now I'm in a breakout room, CPH 2363.
it's very good to know that you spoke with citizens about the services and disservices. My question is, what about the residues from the plants? Do you have a plan to manage the let's say, the waste, the twigs, the leaves, etc.?
Yes, we don't study that. That's not our expertise. But we have a lot of talks about that with city authorities. So they treat it like toxic waste. So they compost it. I don't know what exactly they do it with this, but they have.
They don't do compost or biochar or something?
No, unfortunately not. I only know that they just don't use it again in a horticulture because we really work with the polluted areas. So our plants are very close to the road, very close to emission sources. So when you compost, you also increase the concentration of pollutants.
This is a solution that must be found.
Yes. But we cannot do everything. So this we don't study, but at least I see a huge change in the thinking of the city authorities. So the first step is to make them understand that this is a problem, and then I think they're smart. So we will continue that, and we will find a solution that you can do a circle.
Any other questions?
Thank you, Alex. It's a nice presentation. I want to know that conifers, and if you compare conifers and broadleaf trees, which one you prefer for the better accumulation? And if you use the word biofilters, so it's like accumulation of dust or absorption of dust. These two things are not clear.
About the conifers and broadleaf plants, it's a great and difficult question. The answer is difficult because conifers are better, but we cannot use them, most of them, because they are sensitive. So they are tolerant, but within one season. Longer we keep them in city conditions, close to the sources, easier they are dying. They have some problems. So we use conifers, but only the most tolerant ones. Like the black pinus is a species that can be used. Taxus, Taxus baccata is also very good. but most of them we cannot use. So if we suggest conifers, it's not the front line, it's not the first line, just behind the first line of plants. And also in our location there is very little of conifers because they have no power to compete with invasive broadleaf species like Acer negundo or Robinia pseudoacacia. So when we work with these, let's say, semi-natural locations, there are no conifers. In the city, outside of a city center, when the greenery is planned, we suggest to use conifers, but only where we know that the pollution is not high. So that's why we are looking for other opportunities, like broadleaf trees that keep the leaves for the winter. The leaves are dead. They are not that efficient like in summer, but it's kind of improving the process. Also, we have some species that from their shape, like branches, they are dense enough to filter air. And the second question, for me, all the processes are involved. It's passive, it's active, because plants they are also performing the gas exchange. So all the, I don't care how plants do that. For me, important is that behind the plants, the air is cleaner. All the processes are involved. We believe that biodiversity and a proper planning, I know the word proper shouldn't be mentioned here, but when you know how to plan and locate your greenery, you can really significantly decrease the air quality. Maybe not by 90%, but 50, 60%, like more than half. It's possible. This is what we get from our measurements. And I think when you take into account the whole city, it's significant.
Okay, I think it's clear to you. Further discussion, you can have it from outside. So thank you very much. We have an online presentation by Satinder Kaur from CSER Needy, Nagpur. So Kaur is presenting online. The title of the topic is Developing Novel Air Purification Devices for Removal of Volatile Organic Compounds from Indoor Environment. So Kaur is ready.
So my presentation was scheduled 9:40.
So first of all, I would like to thank organizers for sending me the invitation and giving me this opportunity to present my topic on developing novel air purification device for removal of volatile organic compounds from indoor environment. My name is Satyendra Kaur. I am a senior technical officer in National Environmental Engineering Research Institute, Hyderabad. So VOCs have attracted attention of many researchers and are considered as dominant pollutant. What is VOC? VOC is an organic compound having an initial boiling point less than or equal to 250 degrees Celsius, measured at a standard atmospheric pressure of 101 kilopascal. VOCs are generally classified as very volatile, volatile and semi-volatile based on and are categorized as aromatic, aliphatic, halogenated hydrocarbons, terpenes, derivatives of alcohols, ketones and aldehydes. VOCs are studied extensively. SCPA in one of its studies have detected 900 VOCs indoor. Around 189 hazardous chemicals fall under the category of volatile organic compound. It was also found in one of the as compared to outdoor. Air quality services detected 12,000 unique VOCs from 65,000 different products and that releases VOCs. It includes building materials, room decorations, carpeting, painting products, furniture, fabric materials, pesticide, detergents, newspaper, air freshener, scented products, etc. When we are inhaling these VOC released from these products, some short-term and long-term health effects can happen. Short-term include eye, nose, and throat irritation. You might have observed whenever you buy some new furniture or if you have painted your house, suddenly you feel this eye, nose, and throat irritation. You feel nauseated. You feel headaches. You have vomiting, dizziness, and asthma. That are some short-term, effects of inhaling VOCs. And long-term effect include when you are inhaling it for a very long time. If the cause is occupational, then it may cause cancer, liver damage, kidney damage, CNS damage. So this has raised concern among different researchers around the world, and for last 2 or 3 decades, continuous studies are being done to find out concentration and type of VOCs or different kind of VOCs that is being released from different products and what kind of health issues they are causing. So I have listed out studies and what kind of VOCs and the concentration of VOCs being released by different countries. So these are some of the list of VOCs released and studies done by different countries. They have become very serious and they are taking it very seriously that VOCs should be monitored and some control measures should be we should find out some control measures. I have listed out some very common, products that we are using indoor in our households or in offices, which releases VOCs, and we are in direct contact of these VOCs, such as mosquito coils, paints, floor cleaners, detergent, cosmetics, perfume, adhesives, etc. And these are some common, there are other number of hundreds and thousands of VOCs, but I've listed out few common VOCs that are found in this product. Now, out of these VOCs, I've listed out common, which was found in all the indoor environments, such as offices and households, and the kind of health effects it is having. So you can see toluene or benzene, it causes acute leukemia, bone marrow abnormalities, etc. So how to control these VOCs? I mean, we are using, there are different activities that are happening in our households, different products we are using, but how to control, to have a direct contact with these VOCs. So there are certain control measures. One is, of course, avoiding not buying products that have VOCs or that releases VOCs, or selection of products that have low VOCs. So a few years back, we have done this project by Godrej, where they have manufactured certain furniture that had low VOCs that released for the office environment. So we tested that, and yes, they released less VOCs as compared to other furniture. Formaldehyde is one of the main VOC that has been released from the furniture. So these furnitures, so selection of certain products that releases less VOCs. Then substituting products that releases VOCs with for example, water-based paints instead of paints having VOCs in them. Then good ventilation is required. We keep our homes and offices and school and all indoor environment completely closed because of air conditioning and heating system. So it should be made sure that proper ventilation is there once in a day, so that all stale and pollutant is exhausted out. And then there are certain control devices that are used to control VOCs. These are physical technologies, chemical and biological, but it too has their disadvantages. They produces byproducts. They causes lots and lots of energy consumptions, especially in the developing countries where there is energy is a problem. They are not cost effective. High maintenance is required. Then less treatment efficiency at high concentration. Whenever in indoor environment, if a low concentration of VOCs are there, they will work well. But at high concentration, they don't show that much of result. So I have listed out some of the technologies with their removal efficiency, advantages and disadvantages. So these are the physical one, plasma technology, non-thermal plasma. They have their advantages, but then again they have their disadvantages such as byproducts, as I've already mentioned, poor energy efficiency, etc. These are some chemical technologies in which some chemicals, some products are used to reduce, such as absorption process is used, activated carbons, etc. But again, it has its advantage but disadvantages also. Now, these are some biological technologies and that are being used to reduce VOCs. It also has its advantages and disadvantages. Apart from these technologies, it has also been found that plants are a potential green solution for reducing VOCs. Potting plants, through many experiments, have shown high efficiency in removal of VOCs from indoor. Apart from that, it improves health of the inhabitants, result in a cost saving over traditional whatever technologies I have shown. It is more cost saving, more cost effective. There are no byproducts. It can be reused again and again. I mean, in other technologies, if filters or catalysts they get saturated, we have to replace it, or some other technology is used to clean them. But in case of plant, we don't need to use that, and it has been found that efficiency of plant increases with time. They get adapted to that environment, then work equally well in air conditioned environment and in absence or presence of light, and it improves the aesthetic as well. So these are the, I have listed out some plants. I mean, it's more than 100,000 that they have been tested by different researchers around the world, and you can see it has shown efficiency up to 80 and 90%. So these are the different studies by different authors and different kind of plants throughout the world that are tested to reduce the VOCs, and it has shown promising results. Apart from VOCs, it has been also observed that these plants can reduce PM10, nitrogen dioxide, ozone, and carbon monoxide. A peppermint plant absorbs nicotine from indoor air environment. Plants also reduce airborne microbes present in the indoor air. With the reduction of accumulated pollutant by plants, many health problems such as body ache, headache, dizziness, irritation of the eyes, running nose, dry throat, cough, nausea, skin rashes, itching of scalp disappear. So we also tested some 26 species from 20 genera and 14 families locally available around us. We mostly use them as a decoration, as ornamental plants in our house. We tested those. We took those plants and tested in our lab. There was a experimental setup in which environmental chamber was there with a controlled temperature, light intensity, and relative humidity. Then pots of fixed size and weight was used, in which these different types of plants were taken in triplicates, and then there was a sealed chamber. You can see in the photograph, there was a sealed chamber, completely sealed, in which pollutant was injected, but there was no leakage and no other pollutant can escape or come inside. There was no leakage. So this sealed chamber was used, in which a plant was placed and then polluted was injected. So initial concentration of 120 ppm BTX mixture and then 30 ppm individual, that is benzene, toluene and thiobenzene, were injected. 1 ml of air sample taken using a gas-tight syringe after 6, 12, 18 and 24 hours. And then these samples were tested on GCFID. Then reduction was seen with respect to percentage reduction and leaf area. All experiments have been performed only after leaf test of empty chambers and later with plants. So these were the different plants that were tested for their ability to reduce VOCs. So plants have shown reduction from 80 to 90% in 24 hours. Most of the plants have shown 50% reduction in 4 to 8 hours. When removal efficiencies have been compared with leaf surface area of the plant out of 26 indoor plants, 10 showed highest removal efficiency. So these are the list of plants, top 10 plants that have shown 80 to 90% reduction. About 8 species reduced benzene, toluene and xylene, 12 species reduced ethyl benzene and 9 species reduced BTX in 4 to 8 hours. So that soon they were able to reduce the VOCs. Peperomia caprita had shown consistently a lower level reduction, and Calicut tomentosa and Sensibilla cylindrica had shown highest level of reduction. Results clearly show that most plants have shown higher rate of removal from toluene. Toluene was the one that was easily removed by this plant. Highest removal efficiency was shown by Crassulaceae family, whereas lowest removal efficiency was shown by Pennaceae family. So these are the reduction. You can see plants, they have shown reduction and then it tapered off. All are showing a reduction at different time period and at different rate. But they have shown reduction, and that is for sure. So plants were then ranked based on their reduction efficiency and at what time they have reduced from as poor, moderate, good, very good and excellent. So top 10, the plants that came in excellent or very good category were further used in designing the technology air purification device. So there are certain challenges also by using only potted or individual plant as purifier. Researchers suggest that for maximum removal of indoor pollutant, multiple plant species are required. So from the test results, it was clear that a few plants were able to, were able to remove only certain kind of VOCs. Only very few plants were able to remove a mixture of BTX. And when different products are used indoor, a mixture of or a cocktail of VOCs are being released. So in that case, only one plant is not sufficient to reduce those VOCs. So consortia of plants or different kind of plants should be used in unison with technology to reduce that level, because on daily basis those VOCs are being released. So this study was already initiated by NASA in 1989, and they have recommended that 15 to 18 house plants in 6 to 8 inch pot size to improve air quality in an average of 167.23 meters square of house. In another study, it was suggested that about 680 plants would be needed for a 340 meter cube resident house. Process of removing the pollutant by plant is very slow, and it takes about 24 to 70, and these VOCs are being released on a daily basis. Continuously we are using some kind of product and they are being released, so it need to be consistently removed or reduced from the environment. So what should be done? Future experiments should shift the focus from potted plants' ability to passively clean indoor air and instead investigate biofiltration technology, which uses all kind of techniques, that is physical, chemical and biological, as well as plants, to reduce VOCs. So that is what I've written. Combine all three technology, that is physical, chemical and biological, into one to increase the efficiency reduction, clean more air in less time and develop a device which will require less of space. This is what was said in the study by Wolverton in NASA. And Wolverton was the first one who actually initiated this study on biofilters. So our aim was to combine all the three techniques and make some technology that give more efficiency in less of time and space. So this device was was suggested and is based on aeroponic technique. What is aeroponic technique? Is a technique where plants are grown without soil by suspending their roots in the air, misting them with a nutrient-rich solution. In this case, we are not using any soil media or any kind of growth media or any kind of activated carbon which was used in previous studies by NASA or Wolverton. What we have done is we have simply stranded the plants, and the liquid nutrient is been or solution is been given to the plant. On the rhizobium, microbes grow, and from one end you can see this pollutant is sucked into the device, and very slowly air passes through the roots having rhizobium microbes in the rhizospheric region that helps in reduction of VOCs, and from the other end clean air is passed out. So from the diagram it is very clear, you can see. So this prototype was tested again in a clean room chamber without the trace of any VOCs or any kind of pollutant, and then VOCs was injected in the known concentration of VOCs were injected in the chamber, and you can see at there was a controlled temperature or relative humidity, and air exchange was 27.5. Initial concentration of 30 ppm. Again, we have kept the concentration same. Both low and high concentration was used to find out the ability of plant to reduce the VOCs at both high and low concentration. And then a device was switched on after 15 minutes of mixing of these VOCs, and then this whole test was conducted for 90 minutes. The whole VOCs reduced level was reduced within 90 minutes. There was and this whole sample was tested both with the help of VOC analyzer, as well as samples were taken and tested on GCFID. Then further percentage reduction and clean air delivery rate was found out. So from the test, graph, you can see how the VOC reduction took place, and later on it tapered off. So chambers, you can see device is showing 45 to 52% reduction during 90 minutes of test period, where benzene is showing 51, toluene 52, ethyl benzene 49, and xylene 45%. BTX had shown 49% reduction. Then single pass efficiency of the device was from 44.5 to 51.9%, and the device had shown clean air delivery rate ranging from 10 to 20. This particular device is a novel technique, a completely new idea, and is being patented both India as well as in US. So still a study is going on, and we are trying to find out different parameters and different aspects that can be affected while using plant along with this technology. We have tested right now for 24 hours. We are trying to find out how long the plants and other features can help in reducing VOCs. Then response and tolerance of the plant with respect to the device, response of microorganism present in the rhizosphere, different concentration other than 30 and 120, and at what point the whole device or plants can be saturated. Plants have shown good results. They adapt and show better results with time. So thank you. That was all. I hope I have completed within time period.
Thank you, Kaur. Because of paucity of time, we cut short discussion session. Now the next presentation is also an online presentation by Kim from University of Helsinki, Finland. The topic is New Forest Garden at the University Helsinki Wiki Campus to improve biodiversity and stakeholder involvement. So welcome, Kim.
You can start, sir.
Hello, I hope you can hear me.
Yes, sir, you are audible.
So can you see the screen? I'm a Timir, I'm from Helsinki, Finland, and my talk today is about city planning and how cities can become much more sustainable and be much more efficient in the green transition and also to combat. I tell you about what we have done during 1 year at the campus of University of Helsinki in Viki. So it's about founding a new forest garden at this campus. So, the climate change and diversity loss are the everyday challenge. Yes, we have too much CO2 in the air. The thawing of permafrost, which is already going on, is releasing methane, a greenhouse gas which is 80 times stronger than CO2. Unfortunately, the biodiversity loss is accelerating. And in this situation, cities are suffering from the cities developed into something like with asphalt and concrete, which is an inorganic environment, and that's a dead environment. So urban soil management is fundamental in, or should be fundamental in the development of cities. So Finland is part of EU, and in EU they finally got a sole strategy last year to establish some verifiable national and local targets for reducing land use in cities. And this strategy will also strengthen the objective of no net land take, so that more land should not be taken for building in the cities. And the heat and flood challenges in cities will affect mainly people with lower income and those who have health problems, and especially elder people. So, in the cities, the city planners seem to forget the organic matter. Organic matter, that's the forest, that's the soil, and then we have the shall we do? We can introduce forest gardens. So soil is also very important for human health. In cities considerably, so we should increase the open soil. And generally speaking, so far, very little attention has been paid to the fact that there are favorable microbes in soil and in plant systems that actually affect human health. And this is a topic that was shown already by Finnish, or first time shown by Finnish researchers, the ecologist Ilkka Hansky and the medical doctor Tari Hahtela some 10 years ago, when they showed that actually microbes affect the functioning of the human immune system. So this is about, that's but at the same time we should understand that we need also the healthy soil ecosystem, and that we can get by doing right type of soil management. So last year I was in Paris, and in Paris they have founded microforests in the very center of the city. The term soil sealing is used to refer to covering the soil, and that's very dramatic deterioration of soil. But in Paris, they have opened and tear up the soil by removing the asphalt. Continuing doing so. So they take this kind of a very drastic and, if you would say, old method, doing it really by planting a huge amount of trees in cities. So the trees are lowering the local temperature in the cities. So inspired by that, we last year came with the idea that yes, we could introduce a forest garden at the University of Helsinki Viikki campus. So we started a community project last year, and at the very center of this project is the social function. So our group, we want to involve people to feel ownership to the place where they live or where they work, and also for them to have the ability to experience sense of care or taking care of this case of forest garden. And in this presentation, I will now tell also what the forest garden, actually what I mean with the forest garden. Here we can see the development of the forest garden. So to do it, you need a lot of biomass that you can see in the left-hand corner. Then we were able to involve local people and friends, people who are interested in this type of forestry and doing something for the environment and increasing the biodiversity. Then here to introduce fungi to the soil. So these logs were then covered, like you see in the down in left-hand corner, and they have different types of microbes, wood decaying fungi in these logs. And then also we are planting different types of trees and also edible plants. So this is actually the sign that will be put up at the forest garden at Viki campus. And here you can see a schematic presentation what we have there. So we have raised beds in the middle. The 1A is a raised bed. Then we have a hazel grove, conifers with high bush blueberry. Then we have included fruit trees, and this is on a field. So to the right we have a windbreak with hazel, raspberry, and black locust. And at the just adjacent to our forest garden is the campus gardening area. So already this planting started this year in May, and in about a month, when the flowers that we had planted started blooming, so we could already see how. And I told that yes, soil is important for human health, but we also have the more than human agents, so we can also look at how the organisms or animals at the forest garden are experiencing this new ecosystem that we have founded here at the Viki campus. And in June there were lots of bumblebees in the forest garden. So what's the principle of a forest garden? So it's a food producing forest that's seen to plants that are densely planted, so it's a very productive land use. And the system is universal. You can do this, of course, this forest planting with the same principle all over the world, but it's of course the climate, the local climate and location will decide what types of plants you can use. And the important thing is that everything that you plant should work together, so you make ecosystem where these different plants most temperate forests, like in our part of the world, you can say they have 7 layers. And but if you make a forest garden in a tropical environment, you could have up to 13 layers. So the 7 layers of a forest garden, they first you have the upper canopy. You have the highest trees. They can be also sweet chestnut, for instance. Then you have lower canopy tree. Plants. Then you can have the 3rd layer, you can have vines and climbers like grape. And then in a forest garden you also have shrubs and understory bushes. And then just herbaceous, perennial. And you can see to the left we planted strawberries on the raised bed. And then, of course, you still also have the roots and the rhizosphere for all the plants. So this dense design is really good for carbon sequestration, and it's particularly advantageous in cities where land is scarce and expensive. So this is kind of a intensive land use. And what's really, of course, fundamental here is that you are, with this artificial or new ecosystem, you are also providing ecosystem service enhancement. And there are multifactor benefits, so it's also about food security, so that we should in the future be able also to produce food directly or local food in the cities. A well-managed garden can yield nuts, and in a forest garden, it requires little or no extra energy. And actually, when the forest garden is done and is working, it requires a minimal labor. So plants and insects, but also for animals. And it's plants so that you also have greater pest resilience, and naturally you are not using any chemicals in taking care of the forest garden. And also you don't do any tilling. So forest gardening could also be said that it's part of the wider approach of agroforestry. And as I mentioned, there are several ecosystem services, and the immediate ones, like in Paris, where they planted microforest, is this cooling of high summer temperatures, but it's also a means of pollution mitigation, and above all, looking for people living in the cities. I have also published an indicator in a book called Towards More Sustainable Residential Areas, and I introduced a sustainability indicator, that is ratio between impermeable and permeable surfaces, and that's something that these kind of indicators would be needed for the cities in sustainable development. And this book is available in English, and it was published by the Helsinki Institute of Urban and Regional Studies. So we can start building also in cities, also on poles, and I know it's done in many countries. It's a biological forest biological station in southeastern Finland. So what we our work is done on a voluntary basis, and we didn't have hardly any funding. And what we have now applied for funding so that we can start monitoring this forest garden. So we will monitor the soil and the microbes. We will monitor how the plants are developing, and then we will monitor the insect life in the forest garden, as long as we get the funding, and we will hope to be able to do this. But the important thing is that this has been done completely outside institutional science, outside a university, that it's kind of a thing that we have done outside our everyday work. And we hope that cities could be going towards an organic area and to meet the sustainability requirements. So thank you for this time, and yes, I end here.
Can you unmute that they can hear us? Can they hear? Okay. Thank you for the presentation. Excellent job. We have time for at least one question.
My question is, we can plant as many trees and as many plants we like, but unless there is a reliable source of water and nutrients, they eventually die. So what's the source of water and nutrients for all these plantation, so-called urban forestation, highways, whatever it is? Where is the reliable source of water and nutrients?
Yeah, I really can't hear the question.
My question is, we can plant as many trees and plants. Where's the reliable source of water and
the plans so that you don't, well, okay, when we started this, we had available, water was available, but need actually, when it's growing a little more, it should not need and nutrients, we don't add. What I forgot to say is that we have added biochar when we planted this forest garden, but we are not going to add any nutrients because the way it's planned, it should not be needed.
All right, our next speaker is Dr. Pawan Agrawal. Here the title of the presentation is a cost-effective phytotechnological method for treatment and disposal of municipal wastewater.
While I traveled from Australia, Sydney, Australia to this place, I thought I might go via Varanasi to have a look at the river Ganga. And I collected the water sample from river Ganga. I thought it will be sparkling clean, people taking dip and drinking water and all that. I couldn't myself get enough courage to drink it. This is water which people rely on. To every Hindu, it is holy water. Now, what I'm going to talk is, can this water quality be improved? How? How long will it take? What is the cause of this problem? What can be done about it? That's the topic of my talk. So, the question is, so what I will do is, I briefly have covered background of the problem, concept, applicability, impact and conclusion. Now, if you look at this, young children are having fun in polluted waters, right? Would you believe 1.5 million children and adults die in India alone every year? Are we doing anything about it? We are not. Polluted waters cost families almost $4 billion a year in India. Are we doing anything about it? No. 80% pollution comes from little villages. And people think that we can provide electricity plus water, but third necessity is sanitation. Unless we have sanitation provided by these people, we cannot survive. There are almost, when I saw the problem, I thought I might do some background check or collect some stats, and I found that there are some 649,000 villages in India and 8,000 cities and towns in India, 1,093 treatment plants in the whole of India. So what happens? Wastewater from all these villages and towns flows directly into river, pollutes the river, and causes disease. Can we do anything about it? No, we can't. The reason is, all over the world, the technology being used is called activated discharge process. I have been involved for the last 35 years in India and in Australia as a pollution control engineer. I've designed numerous water and wastewater systems. Is problem? Okay. Water and wastewater systems to design to meet the receiving water quality standards, whether it's ocean outfall, which I did at Boulder Bay, or whether inland sensitive water. This process was invented in 1913 by Arden Bennett in UK. Would you believe, after 120 years, we are still sticking to that system all over the world? And it is costing us $6 billion a year in upgrading the system. And that still we will never reach that 700,000 villages. Oh God, sorry. Okay. So what it means is that the present technology is not sustainable, it is expensive, it requires huge, believe me, I built it, huge civil engineering structure, tanks, digesters, aerators, all that. Later I'll show you a slide which I did. So what it means is a new method is needed, which is sustainable, which is affordable, and which can provide immediate service. Okay, what's happening? Have I done something wrong? And that method is, I will draw the conceptual layout. Later on, I will delve into details. What it is that we take partially treated wastewater from a supply line and then take it through that pipeline, which is I would call it high density polyethylene pipeline, PE pipeline, drill holes into it, take feeder pipe of it, then drill hole into the ground, 2-meter hole into the ground, and discharge into that. Water will percolate, and the trees will take up the soil. Basic concept, not even a single drop of water is wasted. Not even a slightest grain of nutrient is wasted. Everything is taken up by the trees. So, simple concept, but I'll go into detail now. This one. The first part of it, we bring the pipeline in. It's a raw wastewater. How many of you know that raw wastewater from these houses is 99.9% water? 99.9% water. And it has got some floating ones like plastic, rubber, pipes, which is a nuisance for us. And you've got to remove that, otherwise they clog the pipes and pumps. It also has got many people in here also they wash their pots and pan with sand. So sand goes in, sand actually roughens up the pipes. So remove that. But then you've got to introduce some live activated bacteria. Once you do that, it becomes a very self-promoting, self-growing bacteria mixture. And then you take that. Don't take away from this whole thing any waste from the toilets or kitchens or anything, or washing, nothing. Those are the resources which we are going to use. So what we do is we just take it down through the pipe, through these holes, and come back into this. So these holes are, you can see that this is raw trees, existing trees, it can be new trees, doesn't matter. The pipe that comes in, drill holes away from the canopy, away from the root system. We don't want to damage the canopy or the trees or the root system, nothing. So it doesn't have to be very big. It's only 150 millimeter fiber. This big. That's what it needs. You don't need that big size. No, only this big. 150 millimeters. You have done it. Oh, God, what have I done? So, these feeder pipes are only 10 millimeters. That's size. So, if you look at the holes, these are only 300 diameter holes, 2 meter deep. We have got the idea from electricity poles. They drill the hole, put an electricity, and that's it. We could use the same machine, same technique, and he could drill, believe me, he could drill 100 holes in a day. It's so efficient. And we tap it at the bottom so that we don't drop our mobile phone in it. Now, what we do now is the next one. We got the pipeline coming in, pipeline, put a feeder pipeline in, water, this is that what we go from. Okay? What happened now? Some of the heavy metals, you can say that the pharmaceutical residues from our bodies, from endocrine structures, and all the you can say things which you don't find in the satellite water, in the satellite layer. But the upper part of it is water, wastewater, with all the bacteria in it. What happens then? They are trying to contaminate the surrounding soils. When the surrounding soils, they are the food for the soil microorganism. They microorganism, they normally just stagnant, no food, no water. But the moment they get that water and nutrients and food, they come into life. Their characteristic is that they can convert chemical compound into plant usable ionic form, nitrates, phosphates, whatever. But they also carry water. Water is nutrients still try to absorb or cling to the little pore spaces and become an ionic form. Wait till somebody asks for it. The water will keep on percolating and eventually, may not, eventually end up into a little creek or pit or whatever it is. May not happen, but if there is any surplus water, that will go in there. But by that time, it has been filtered out fully. It has taken care of all the pathogens, nothing left, and believe me, it oozes out like pure water. Okay, so what happens then? So the food goes away from the roots and benefits. So the trees, because they got 2 meter deep holes, the trees will develop deeper roots. That gives them better anchorage. That gives them much more vital source of water and nutrients. What also you get, the soil gets enriched this entire area. So they're not concentrating on a small area, but gets enriched. The sediments layer, that also got a function. Because all the heavy metals from here, they are useless otherwise. But later on, they could become useful. So how do we get the water and nutrients from here to the tree roots? It's done by our biologists, but 100%. If I put it in they transfer it to the tree roots. So let's look at what the tree does. tree needs for photosynthesis water, some phosphorus, sunlight, and yeah, and carbon dioxide from the atmosphere. Yes. Carbon dioxide from the atmosphere, and it converts it produces both carbohydrates, sugars, also lipids. It uses almost 65% of it produced for its growth in its upper part, subsurface part of this, whether it's fruit or leaves or whatever it is. 35% of that part goes into the tree roots. When it comes to tree roots, all those sugars, they exude from the root tips. And there we have got fungi waiting for it to come out. They immediately grab it. And when they grab it, they start growing. These hyphae start growing. And the mycelium gets elongated. It has got wider reach. Why the trees do that? Why the tree feeds this fungi that much of sugars which it can use? No, because it needs the nutrients and water from a wider area. Tree roots can only absorb from the root tips, but fungi can absorb nutrients from the entire length. And that's where characteristic. That's why trees encourage fungi to grow so that they can get, so when the tree needs water, it asks the fungi, come on, give me some water. And the whole lot of them, they know that they survive on from food from the tree roots, they survive water. Tree said, I want nitrates, they provide an ionic form of nitrogen, phosphorus, zinc, calcium, whatever they needed. Let's say that we are growing a tree, fruit tree, let's say mango tree. Now it might need certain more vitamins, right, which we don't, soil doesn't have it. So the micro is like, we will go down into that area where there are some of those vitamins we consume that lying there. They can actually, because it has bacteria there also, they're mineralizing it, they take it up and they transfer it back to the root. So what happens? We have got, the trees have got plenty of water, plenty of nutrients, trees grow. And so what happens is, you can see that as circular system. We consume food, transfer wastewater, tree goes, comes back, a circular system. Right? That's in water, nutrients, phosphorus, everything. So, what it is, that's like, if you look at it, this is how I was trying to show that fungi actually take it up, penetrate into the roots, and then they actually transfer it like a that goes inside the root and then blow out and then finish. The tree, in other words, gets lots and lots of nutrients quite easily and rapidly. So, look at it why. It's a simple concept. It's no mechanical, no electrical, no involved. It is such a stark departure from the present practice of sewer treatment. Believe me, I've spent 35 years in designing a number of sewer treatment plants from start to conclusion. How this concept came in? I was addressing public meeting in Australia. Very Irish public meeting. One part of what I said, hey, why can't you feed these trees, feed this wastewater to trees so that we can grow food? I have no idea, believe me. I simply said, I'm sorry, Mike. In Australia, we don't want to start. One is not mine. I said, sorry, Mike. I don't have that. You couldn't get the answer. I couldn't tell you. That's how we got to read for a long time. I joined. That's how I told you that before. The formal course, 24 minutes, I studied that. And that lecturer, I said, I'm looking for an answer. Whether wastewater can be used to build the trees. They said we have never done it. So combine the knowledge of my new knowledge, experience and all that, with all these lecturers giving inputs from new fellows like that, combine it and this concept emerged out. And it's so simple. I said, God, So what it does is, you can run it. It provides sanitation facilities at lower cost and lower operating cost. There's no cost, no gravity required, not much anyway. And then it can be smaller scale. It can be from what? Small one room, one house, 200 houses, 2,000 houses. Two cities. Cities are of course made up of the world. And it can be still okay. It can grow trees. Where do you grow trees? You just talk about growing trees for space purification along the highway, along the routes, along the anywhere, parks. They talk about where that gentleman talked about the urban forestation. And I said the same question. Where is water coming from? Where is the nutrient part? This provides the source. And you can use that, and we have done it. Along with them there is a road, there's a road reserve, which nobody can do anything, but you can grow trees. And then, and we can use all types of trees, fruit trees, like for air purification trees, small, larger can be small, can be matter. So in other words, if you've got a village, right? I've got a village, there are trees, you can grow the trees, put the ball in the, drill the holes in the ground, feed it, done. Engineers love to do the design first and estimate. So I said, okay, if I have to design it, how much it is? Each household produces about 500 liters a day. And let's say, so what this means, we feed 10 trees, right? So we can say, okay, per house, 10 trees. How many boats will we need? 5 boats per tree. So for small houses, this is the design. For small houses, one house, for 100 houses, we produce 20,000 liters of wastewater. It will need 1,000 trees, big, small, bigger, tiny, whichever way you want it, both sides of the road, 2 rows, 3 ways, whatever. And you need 5,000 holes, easy to drill, within a month you can drill it. And electrical pipeline is 5,000 meters. And I know the Australian estimate, but actually it is not very expensive. We're not talking this pipe, we're talking only this pipe. Believe me, this pipe. No, and very little pressure pump required.
Sorry to interrupt, Dr. Kavam. We have only, your time is up. Can you finish in
Okay.
3 minutes maybe?
Okay.
Thank you.
So you can see the impact, positive impact, reduction bottom, waterborne diseases, revenue-free crops, environmental, you can healthy free carbon. And this is not a date, you can say traditional method, there are 15 steps, 15 structures. Right? I did that and then I didn't show the other one. All these mechanical structures will lead to small ones. And so you can see that you can evaluate the two processes. So, my point is this, there's an urgent need to reduce human suffering. That is the guiding principle for me. And this phytotechnological method is affordable, sustainable, technologically reliable. Thank you.
Thank you very much for your impressive presentation. And indeed, it's very important matter. And we do have time for questions. 1 or 2 questions, anybody? I do have a question.
Yeah.
You mentioned about so the whole process, and then the fact that there are pathogens in water. Have you or do you know of anybody who has tested water after going through the process of phytotechnology for pathogens?
Yeah, okay. The answer is we have not tested it. What we did was that I didn't show it. The place called Nabaka Hats in New South Wales, the EPA drove us to the wall saying that you cannot discharge into a waterway. So discharge into a forest, and what happened was that we didn't expect unintended consequences. In that area where the water went, the trees start growing taller and taller. And the creek where it was going into, right there were people swimming in it. We kept our record from the hospital. Are there any incidences of waterborne diseases coming to your hospital? And it's a reportable disease in Australia anyway. So we kept on track of it, like organic people, you can't stop people from swimming. So that's what, that's the only proof. We have not done, we haven't done any laboratory tests, no.
That's valuable. Thank you very much. Any other questions? All
Want
right.
to ask question? No, that's okay.
Oh, all right. Thank you very much.
Thank you. Anybody want water?
But not least, presentation is also online. My apologies in advance if I pronounce the name incorrectly. Lizivi Mogivisa, and the title of the presentation is Heavy Metals Immobilization of Mine Polluted Soils for Cultivation of Saponaceae oleracea using Saccharomyces cerevisiae suspension, so using fungi for phytotechnology. Oh, mine, interesting, thank you. Just be mindful about the timing. We keep it hard on 20 minutes, and that should be done by 17 minutes. The presentation should be over. Thank you.
Can I start? Okay. Thank you and good morning. My name is Liziwe Mukibisa, and I'm from South Africa, from Sefako Health Sciences University in Pretoria, in South Africa. My presentation is on the heavy metals immobilizations of mine polluted soils for cultivation of Spinacia oleracea using Saccharomyces cerevisiae suspension. Next slide, please. Anthropogenic activities such as mining and also urbanization, industrialization, and changes in the land use such as using fertilizers and pesticides contribute to the increase in the levels of heavy metals in the environment. These heavy metals in low concentrations are beneficial for the growth of plants as well as the development and for cell function at biological, chemical, and molecular levels in human beings. However, some heavy metals such as mercury, lead, cadmium, and arsenic are toxic even at low levels. These heavy metals cause diseases such as cancer, birth defects, kidney dysfunctions, as well as the pulmonary and immune dysfunctions. Next slide, please. The challenge is that the heavy metals are non-biodegradable and persistent in nature, and this contributes them to remain in the soil as well as in the environment, contributing to their levels being increased in the environment. And also, when these heavy metals are accumulated in the soil, they can actually be transferred or be taken up by the plants, and humans who consume these plants end up accumulating these high levels of heavy metals, which are transferred through the trophic food chains. As a result, the consumption of food crops with high levels of heavy metals, which are above the regulatory standard limits, is harmful to human health. Hence, there is a need for the remediation and immobilization of heavy metals in the soil to prevent the uptake of these heavy metals by the cultivated plants, which are consumed by the human beings, and these actually contribute to food security. Next slide, please. However, the application of bioabsorbents, which can contribute to the immobilization of the heavy metals in the soil to prevent them from uptake, these are bioabsorbents such as the red mud, the manure, mature, reduce the availability of these toxic heavy metals to plants. However, they change the physical chemical properties of the pH of the soil, and can ultimately affect the growth of plants. Furthermore, these bioabsorbents such as bioarcher is that it can only be effective for fewer heavy metals in the soil. Hence, there is a need for microorganisms such as Saccharomyces cerevisiae to immobilize the heavy metals in the soil or from contaminated surfaces. The advantage of this Saccharomyces, which is an yeast, is that it is a promising and valuable approach that is inexpensive, also safe for human consumption since it is generally used for the production of most food products which are edible. Hence, the aim of the study was to assess the heavy metals immobilization of mine-polluted soils for cultivation of spinach using the yeast suspension. For materials and method, initially the yeast suspension was prepared by adding 12 grams of the active dry baking yeast, which is usually used for baking. This yeast was dissolved in 1 liter of warm water, which was at 38 degrees Celsius. This was followed by adding 12 grams of sugar, and the ratio that was used was 1:1. The soil samples which were used for the cultivation of spinach were collected around mining areas. And these collected soil samples were placed in pot plants with measurements of 12 centimeters by 36 centimeters. Next slide, please. After the collection of these soil samples, they were treated with different quantities of the yeast suspension, and the different suspensions were for treatment 150 ml of the yeast suspension was added, and these were the different quantities which were used for other treatments of the soil. Followed by the treating of the soil with yeast, then 3 seedlings of spinach were transplanted into each of the pre-treated pods. Plants were grown to maturity, which took about 9 weeks, and at full maturity, the plants were then harvested, separated into different components, which included the stalks, the leaves, as well as the roots. And these were digested, and also that was followed by the analysis of the heavy metals using ICP-MS. The transfer factors to determine if the trace metals were transferred or only remained in the soil, we made use of the equation, which was transfer factors of concentration of these in the different plants divided by the concentration in the soil. The statistical analysis was done using analysis of variance. Next slide, please. With respect to the results, we have table 1 there showing the levels of the heavy metals in the soil which was collected around the mining area. From the results of these levels, we can see that the heavy metals of these soil samples collected from around the mining areas before treatment was, the levels were quite high, and they ranged from 3.09 to for selenium to about 4,201 micrograms per liter for chromium. When these levels of heavy metals in the soil were compared with those which are recommended or acceptable by the World Health Organization, we found that all the parameters were, however, below those limits. With regards to the levels of these heavy metals in the roots of the plants, which were harvested from the soil, which was treated with different quantities of the yeast suspension, we saw that in the roots, the levels of selenium were even below the detectable limit, and of all the heavy metals that were measured, zinc showed the highest levels, and it ranged indicated there on the slide. The levels of vanadium and level were significantly lower in the soil treated with the highest levels of the yeast suspension as compared to in the control where there was no treatment of the soil with the yeast suspension. Next slide, please. However, in the stocks, it was discovered also that most of the levels of these heavy metals were actually much lower. We also, again, in the treatments where there was the most treatment with the yeast suspension. Next slide, please. Again, in the leaves, the manganese, sorry, in the leaves of the spinach, manganese ranged from 204.9 to 1,556.14 microliters. The importance of manganese is that it serves as a cofactor for numerous enzymes. So it was actually good that the levels were high as it plays an important role. Even though the prolonged exposure to manganese can actually result in neurological diseases if the levels of this manganese in the consumed vegetables is above 5 milligrams per per meter squared, that squared. Next slide, please. In conclusion, okay, sorry, with regards to the transfer factors, it was discovered that all transfer factor values were below 1, indicating the unavailability or exclusion of these heavy metals from the soil to different parts of the cultivated spinach. Next slide, please. In conclusion, we saw that the concentrations in the roots, the stalks, and the leaves of the spinach were found to be within the allowed levels for safe human consumption, indicating the heavy metals in the soil were immobilized using the yeast suspension and hence were not taken up by the spinach. The heavy metals failed to bioaccumulate in different parts of the spinach. Hence, the use of the yeast suspension, and in this case, the use of microbial resources such as the yeast suspension, can be used as the visible and a cost-effective and efficient way to bioaccumulate heavy metals from the contaminated soil environment, hence preventing their uptake by the cultivated plants. These are my references, and thank you very much.
Excellent. Thank you so much. There is 5 people.
Okay, can the organizer please help me going back to the slide?
That's fine. And what are the units here?
No, these are the transfer factors. So remember, we will divide with the same units above and below. So there are no units for the transfer factors. But what is important is that we had to check that they were below 1, which indicated that the heavy metals were not bioaccumulated by the plant.
Thank you very much. you, everybody. So that was our final presentation for this session. I hope everybody learned some interesting material. So we have time for coffee and tea. Then we'll come back here 11:05. Thank you.
Thank you, everyone.
Pleasure to introduce.
Sure, sure. Okay, fine. Okay, so good morning, all of you. Now we are starting this session, which is focused on phyto monitoring, land restoration, and indoor pollution. So, our first speaker would be Mariam El Lamy from Missouri State University, USA. Can we connect? Yeah, Mariam El Lamy. Okay, the presentation will be on the topic enhanced restoration of mine tailing, role of abrascular mycorrhiza fungi and the waste products by the sustainable revegetation and soil building.
To go. There are speakers here.
There is a noise. There is a noise. Could you please reduce that noise near you? Hello?
Yes, ma'am, we are ready to go.
Okay. Hello, everyone. My name is Mariam Elami, and I'm a postdoc at Missouri University of Science and Technology. I'm presenting today my work, which is a continuation of my PhD studies. It is involved some phytoremediation experiments of mine tailings. So the mine tailings, as you know, it is a global issue. Hello? Okay. Sorry about that, but there is noise in the background. Now it's fine. You all can hear me?
Yeah.
Okay, perfect. Yeah, so as you see, this is just within the United States, and actually just within the area where I live in Missouri. There's many sites of mine tailings and associated with legacy mines, specifically. And the problem with these tailings, they cannot sustain any vegetation, natural vegetation, and they are just posing a continuous risk to the environment and to the surrounding ecosystems through wind-blown dust, leaching of metals, actually what you see here, the picture, it is from the site. This is in Viburnum, Missouri. Where I did all my experiments. And it is typical of so many sites in the region. And just to give you some idea about the extent of the problem, only within the US, there is about 500,000 of these sites needing remediation. And the problem with conventional remediation methods, usually they are costly, especially when you are dealing with large scale sites. For example, the sites I'm dealing with is considered smaller compared to the others, and it is only, as I said, it is 270 acre, and it is considered smaller. There's much bigger sites. So conventional remediation is costly, also not environmental friendly. So alternatively, phytostabilization, which I'm focusing on within my studies. Like using the plants, and as I said, I'm specifically focusing on phytostabilization, not phytoextraction. Like using the plants accumulate high concentration of metals, but only within their root system, with very minimal transport to the above ground system. And also with the help of soil amendments, some chemicals to change the bioavailability of metals and rendering them within the rhizosphere, within the soil. And as such, we would achieve physical stabilization with the plant cover itself, with the plant roots, and also chemical stabilization with the amendments. And ultimately, with the vegetation, we could restore the ecosystem in the area. And the overall goal is to sustain soil agronomic properties improvement and soil building. So that experiment I'm going to talk about today, it was based on my previous experiment, which involved the use of soil amendments, as you see here, with a combination of biosolids. We found like the biosolids as a primary amendment is essential to sustain, to support plant growth on these tailings. However, the combination of other amendments, such as biochar, as you see here, further reduce the metal availability within the soils. As you know, biochar has specific characteristics of retention of metals and also nutrients. But the problem that was on biomass crops, the experiment, and was it's only 8-month study, so it is considered, we can say it's considered short-term experiment. The problem, the same site I'm doing this study on, which I showed you early, the company tried to revegetate the site on their own. That was in 2011. However, after 3 years, all the plants, and it was miscanthus grass, died. Why? Because they did not consider a proper modification to the soil characteristics. So here we asked the question, can we sustain plant growth further? So that led to the current study. Can we sustain the plant growth and also promote maybe soil building with the addition of soil amendment, like biosolids as a primary amendment, biochar due to specific characteristics of retention, and also compared to biosolids, biochar, tend to be recalcitrant in soil, so resist decay for many years. And also with the mycorrhizal fungi, mycorrhizal fungi can benefit both plant and soil in so many ways. So, and can also with time promote soil building, like stimulate soil function processes and stimulate soil building. But we need to run this experiment for long term, which we did. And that was the setup for my experiment, as you see here. We tried to only amend the top layer. As you see here, we had 4 treatments, and we only amended the top layer. We used, because tailings by itself, based on my previous studies, could not sustain plant growth longer than 2 months in the greenhouse. So we added biosolids as a primary amendment, and then biochar, hopefully to sustain the benefit of biosolids. And we wanted to test the combination with mycorrhizal fungi, like a specific culture of mycorrhizal fungi. It is isolated from prairie lands, from Kansas City, within the US. And also, in this experiment, we wanted to test native vegetation, like focusing on prairie plants, like grasses, and also legumes, like nitrogen fixins. And actually, this experiment, we ran this for over 6 years. we wanted to test the long-term feasibility of our approach. And the goal was, are we going to sustain plant growth? And also, are we going to sustain the benefits and soil functions beyond the amended layer, like within the soil profile? And after 6 years of growth, then we tested the microbial community as an indicator of soil building. We analyzed this by analyzing for phospholipid fatty acid analysis. So as you see here, a phospholipid molecule are essential component of plant, of cell membranes, and can give information about active microbes within the soil profile, and considered also linked to essential functions within the soil, compared to 16S RNA. It is kind of timely, less time consuming, and also even more cost effective. And the most important thing is it is associated with living microbes, and also with soil, can give information about the components of microbes that is associated with functions of the soil. So here what we got. So this is the total BLFA, as you see within our treatments. So here only like biosolids treatments, and here I want to mention like biosolids added at a rate of 3% and only to the top layer, and biochar at a rate of 2% and only to the top layer, and plus the culture of mycorrhiza fungi. So as you see, there's significant improvement in total BLFA with our treatment compared to just BS, only when talents treated BS. And here actually, you can see here how we sampled for this, and we wanted to analyze within the depth of soil, because we want to see, are we stimulating soil function within the depth? So we sampled in the top layer, and we also sampled here within the unamended layer, and sampled this to be the middle, corresponding to the middle as you see here, and one the lower. So it is, as you see, it is more pronounced, the impact of our treatment, especially with the addition with the mycorrhiza fungi at the very lower layer. That was interesting. And we think that is associated with the root. As you see here, we have root bound here. So the root acted as a vector for microbes. So this is for that corresponding to that part. This is BS plus AMFA at the lower, just from the lower sample or the lower layer, whereas this from the only BS. And you can see the huge differences between root architectural within these layers. Now, just the component of that BLFA, if you look here at gram negative bacteria by treatment and depth, and you know this group of bacteria associated with essential soil functions such as organic matter decomposition, nitrogen cycle, nitrogen fixation, like and other important soil functions. So what we can see here, big improvement, especially in the lower layers, like middle and specifically in the lower layer. And here is the actinomycetes by depth, and as a group of the gram-negative bacteria. And as you see again, just significant improvement in that within the lower layer caused by the AMF. Now, the AMF itself, So as you see here, this is the AMF, BLFA associated with the AMF, and how our treatments impacted that. So as you see, obviously here, the addition of IMF impacted the top layer significantly, compared to the other component. But also, if you look here, we go to the lower layer, there is also a huge impact of IMF, like spreading to the lower layer. Now, this study, it is in the greenhouse, and we are, it is within the pot. So we had root bond issue. However, in the field, the benefits are expected to be different. And actually, for example, that plant species, just I showed you in the where we done the PLFA analysis, it was purple prairie clover, which is nitrogen fixing legume. It is native to Missouri. But it is thought it is ecologically a valuable plant species, and under field conditions it would root to up to 2 meters. So the situation will be different. And actually last year we translated our greenhouse findings to the site. And as you see here, we started this site, and this is what you see here, lines of infected plants transplanted to the sites, and we want to see and study their impact on neighboring plants, the spread of mycorrhiza fungi, and also within the depth of soil. And that, what you see here was by the end of the growing season, and we found great and diverse native plant community, and some of the species, as you see here. So what is next on this study? That species was just one out of 5 we done within that experiment. And also we tried 2 different cultures of mycorrhiza fungi. The other one originated from Great Lakes, from low nutrient origin. However, we were limited in time with the analysis, but our future plans to analyze for this as well, and then link these to the physicochemical correlations. The properties like pH, OC, organic carbon, are we sequestering carbon, metal availability, aggregation stability, and also comparison to 16S RNA data, and ultimately the implication for soil building. So yeah, with that, I would like to thank my supervisor, Dr. Joel Birkin, and my lab mate who also contributed to this work, Gabriel, and Dr. Michael Eze, Dr. Dave Westenberg. And of course, a special thanks to people who helped with the PLFA analysis at Missouri Soil Health Assessment Center. And of course, this work wouldn't have been done without undergrad help. And this work was partially funded by Doran Company. And with that, I'm open to any questions, and thank you.
Thank you, Miriam, for a very interesting presentation. So we have time for some discussions and questions. So if you have questions, you are welcome.
Is it audible to you? Hello, Mariam, is it audible to you?
Type the question.
Hello.
Yes.
Yeah, Miriam, actually.
Species, you mean of mycorrhiza?
Yeah.
Or, yeah, this actually, if you remember, I said there is another, what I tried in this experiment, two mix of mycorrhiza. And each mix contain about 7 species, and they are different species. The one originated from rich soil, rich in nutrient soil. There are 7 different species in it, and the other one there are 7 completely different species in it. And like I'm still testing, you know, these species, species, like the impact of the other. As I said, we were limited with the analysis of PLFA, but the other one, based on plant responses, the growth, it's also what we say, promising. But also keep in mind, if you see, I can go back and show you. We inserted here also a rhizome sampler to collect soil pore water over time. And I've been collecting that soil pore water and been analyzing for nitrogen, for metal, bioavailable fraction of metal. And I found within using those two different cultures, both promising the results I'm getting so far. And actually 2 species of the plants, I tried 4 legumes and 1 grass, because I said it is five different species. The legumes, 2 of them, they showed like signs of nitrogen fixing based on my analysis of soil pore water compared to the initial stages. So yeah, going back to your question, those I have different species, 7 of them. Like which one causing that impact? We still have to answer that question. As I said, we're going to do more analysis, and hopefully we can answer which one. And also maybe we need more experiment, because as you know, with the mycorrhiza species, some of them. Yes.
Yeah, thank you very much.
Availability compared to acidic tailend, but also here you have a problem with the nutrients not available for plant uptake.
Yeah.
And also, as you said, this is not optimal pH for microbes, including AMF. But with the addition of biosolids, actually it reduced that to neutral, around like 6.97, something like that, with the addition of biosolids. So it's biosolids helped with that.
Okay, thank you.
And actually here I want to note the site where I did this site where we translated our finding. Actually, this is acidic site. So pH here around 4. So we had to adjust with lime and to bring that pH to favorable.
Okay. Fine. Thank you.
And sorry, it is too late here. It is around 1:28.
Yes, so our next speaker is Pietro Pieroni from University of Bologna, Italy, and his subject is combined use of root biostimulants to promote Miscanthus giganteus rooting capacity and phytomanagement potential. The stage is yours.
So, do you hear me? Do you hear me? Yes, I think yes. Okay, just to check. Okay, so good morning, everybody. I'm very happy and glad to be here today in front of you and presenting my work that is entitled Combined Use of Root Biostimulants to Promote Miscanthus giganteus Rooting Capacity and Phytomanagement Potential. So, this work has been conducted in the framework of the GOLD project, which is an European project founded by the European Commission. Our consortium is made up of 19 partners from 11 countries in 3 different continents. And especially in the first work package, we conduct plant trials, testing 4 different energy crops to produce biomass for clean biofuel production on contaminated sites. And we test these 4 crops, sorghum bicolor, cannabis sativa, which are annual crops, and Miscanthus giganteus, Panicum virgantum, which are perennial crops, lignocellulosic crops, on 8 different experimental sites to evaluate their biomass production, their phytoextraction capacity, so the potential metalloid accumulation in their above ground biomass, and their phytostabilization capacity, so the possibility to reduce the metal bioavailability in the soil. To increase these performances, we also apply 3 different biostimulants: mycorrhiza, protein hydrolysates, and humic substances, both singularly or in combination. Indeed, our starting point has been pot trials. We have conducted several pot trials, one for each site, collecting the contaminated soils, and testing all the combination of biostimulants over the 4 crops in order to select 2 potential biostimulants very promising to be applied for each crop in field condition. And especially the combination of humic substances and mycorrhiza, the MB2 treatment, was selected by all the partners for all the crops. And considering our results that we obtained in our pot trials at the University of Bologna, we have seen how this treatment could increase plant dead weight and metalloid accumulation in sorghum, hemp, and miscanthus. So, the objective of the study that I present you today is testing this MB2 treatment in the establishment phase of miscanthus. The establishment phase of miscanthus is basically the first year of miscanthus plantation, but can last even up to the 3rd year. In this phase, miscanthus doesn't produce so much above ground biomass because it invests most of the energies in rooting. And when he finishes this phase, it can produce very high yielding of biomass, above ground biomass. So we test this treatment to increase its rooting in this phase and to affect metal uptake and bioavailability to different degrees at different soil depth. We collect the soil from a former illegal landfill. This soil has a sandy loam texture, is subalkaline, it presents a considerable amount of limestone and of stoniness. And the 4 metals are considered contaminant in the area: nickel, lead, and most importantly, copper and zinc. But the starting viable fraction as ammonium nitrate extraction was actually not very high. So this trial has been conducted in an controlled environment in a dedicated structure made up of 10 cylindrical rhizotrons of 66 liters volume each, filled of soil, where the MB2 treatment and the control C were fully randomized. And we considered in each rhizotron 2 different layers of soil, 45 centimeter depth each. And we consider these layers to record hourly the soil moisture and then to sample soil, roots, and soil pore waters. Indeed, we have provided periodically irrigation in the rhizotrons to restore 90% of the water holding capacity. But due to percolation, in the first layer we had a drier soil than in the second one. On average, 46% of the water holding capacity compared to 76%. The trial lasted 7 months to cover most of the growing season of Miscanthus. And in the end, we collect many data about biometric traits, root and shoot yields, and genome through EPCMS, and the metal bioavailability in the soil at the end of the experiment with two techniques: soil per water content, as just seen in the previous presentation, and 1 molar ammonium nitrate extraction. So, what's about the results? We have detected an effect of the treatment in increasing some biometric traits of Miscanthus, especially the total number of tillers of the plant and consequently the total number of leaves. However, these results did not lead to a statistical difference in both fresh and dry above ground biomass. But this is quite normal because Miscanthus was in the establishment phase. It was putting all his energies, his nutrients in the rooting. And indeed, looking at the root fresh density, we have found an interaction between treatment and layers. As in the second layer, in the deeper layer, we have found the highest value for the MB through treatment and the lowest value for the control. While considering the most important root dry density, we have found a very strong effect of the MB through treatment. This effect was mainly due to the increase in root water uptake efficiency, because Miscanthus treated with the MB through produce on average 2.5 grams of root per liter of irrigation provided, compared to the control. And this is probably mainly due to the chemical structure influences of the humic substances extracted from Leonardita that we have applied. These substances are made up to 75% of hydrophobic humic acids. And these humic acids can have a double effect on plant rooting, a physiological effect, but also a physical triggering effect, which we consider most important in our case, through the formation of humic fouling at the root cell wall pores. This fouling increases the hydraulic conductivity in the first part of the growing cycle of Miscanthus, and then increase the root water uptake efficiency in the second part, with the results of the increased root proliferation. Moreover, primary root dry weight, so primary roots, not secondary hard root weights, is attributable to the oxygen and alkali structures typical of aromatic humic acids. Oh, what's happened? Can you help me? Thank you very much. So, talking about metals and the root metal concentration, we have found an interesting interaction between treatment and layer for nickel and zinc in the root. As in the control plants, shown higher metal concentration in the top layer and lower metal concentration in the deeper layer. While in this case, the MB2 treatment acts as a buffer, stabilizing the metal concentration along the soil profile. And it's quite interesting that we have found this similar correlation for nickel and zinc, because we know that are two metals that are subjected to similar dynamics in their uptake in the soil, especially the strong influence of the subalkaline pH. So we have detected even an increase in the estimated accumulation of root. Okay. So what about the metal bioavailability in the soil? The metal bioavailability in the soil at the end of the experiment detected as ammonium nitrate extraction. In this case, we did not record an effect of the treatments applied, but just of the layers. Indeed, lead and zinc ammonium nitrate fraction were reduced in the second deeper layer. But this is due to their precipitation, but with two different dynamics. Indeed, for the zinc, we can assume a very important role of the pH and the increasing moisture in the second layer, which could cause its complexation as a carbonate and precipitation. While for the lead, there is a very important role of the iron plaque formation. The iron plaque is a coating of iron oxides that can be formed at the root surface of Miscanthus and can chelate cation, especially lead. And indeed, looking in detail just at the data of the second layer, we have found a direct correlation between root lead concentration and dry root density. Then, talking about the soil per water concentration, metal soil per water concentration, we have detected several metals, but just focusing on the metals of our interest, we have seen how the MB2 treatment reduced the bioavailability of the copper, underlying how these metals have a very strong affinity for the humified organic matter. But these data are different from those that we have found for the ammonium nitrate concentration. And the reason is basically because in our condition, characterized by, let's say, a coarse structure and a subalkaline pH, the ammonium nitrate extraction can contain a fraction of colloids, metal organic complexes, and metal amine, which could be mask the effect of the MB2 treatment on the soil pore water content, which contain only the most desorbable fraction of metals. Indeed, looking at the multivariate analysis that we have conducted on metal bioavailable fraction, we can see that there is this big cluster in the first quadrant in which metal ammonium concentration in the first layer are directly correlated with their soil pore water content in the second layer. So probably a top-to-down desorption process happened of the readily desorbable fraction of the metals. No other direct correlation have been found in the same layer apart of nickel for the two techniques. But I want just to spend a couple of words about the plant morphological development multivariate analysis, just to show you how the first principal component is strongly driven by all the parameters for which an effect of the treatment has been found, underlying how this treatment helped the miscanthus to perform a behavior which is very well known for C4 perennial grasses to cope water shortage in the first layer. That is increasing the root proliferation in deepest layer, increasing the water uptake, and even the differences that we have seen for tillers and leaves can be a plastic adaptation system to increase the evaporative surface of the miscanthus and maintain the stay green genotype. So in conclusion, growing miscanthus on the tested soil could be a potential suitable phytomanagement strategies to couple biomass production with phytostabilization. And the application of the MB2 treatment, so humic substances and mycorrhiza, could increase its rooting, its establishment potential in this landfill or brownfields or adverse soil, reducing the copper soil pore water content starting from the first year. Other findings is that in phytomanagement system with these deep-rooted rhizomatous crops, some important dynamics in metal bioavailability can occur even below the topsoil, which is usually the most considered, and that the metal ammonium nitrate fraction and soil pore water content measure two different shares of metal bioavailability in the soil. Of course, these findings are related to our experimental conduction. It was a controlled environment with a preferential flow of the water and have to be confirmed under field condition. This is what we are doing. We are almost at the end of a 3 years trial, and I hope to be here soon to present my future result. And just 1 second to acknowledge the co-authors of this study, my associate professor Walter Zegada-Lizzarazzo, my full professor Andrea Monti, most important Dr. Michel Mensch, which helped us to conduct and organize this trial. And I want to acknowledge all the partners of the Gold Project. And please visit our website. Thank you for your attention.
Thank you, Petru, for the nice presentation. So now the session is open for the questions from audience. I invite the questions from audience.
Okay. Yes. There is no space for biochar in your experiments?
Maybe in the future. For the moment, we have focused on this kind of biostimulants because was apparently very in the front of the research. Some research has been conducted, but for many of them, especially humic substances and protein hydrolysis, there is so much to understand, and there are not so much data about biomass crops. Most of the biostimulants applications are conducted on commercial grain cereals such as maize, wheat, barley, rice. So it's very interesting to testing them even on the biomass crops. But maybe in the future we will use even biochar.
It was a provocative question, but thank you.
My question is, out of all these metals like nickel, zinc, copper uptake, after this kind of association, which one we found that it is maximum being uptaken by the plants?
Sorry, can you repeat that?
I mean, you have evaluated all the metals also,
Yes.
okay? Zinc, copper, cadmium.
Yes.
So which one you found most translocated inside the plant?
In the above ground biomass?
Yeah.
Zinc and copper. Especially zinc, but even copper.
But if we consider lead, what about that? It is more toxic for the restoration we require, the lead. to
The
be
lead?
taken up? Yeah.
Basically, the lead is not very transported in the above ground biomass because miscanthus is an excluded species. So it tends to chelate in this iron plaque on the root surface, and it can translocate very small amounts in the above ground parts. I'm talking about 1 maximum to 5 ppm.
Okay, fine. Okay.
Just a short information for the audience. The outcome of the long-term experiment on contaminated site is that biochar alone is less efficient than biochar combined with 5% of compost. And the quality of compost, the quality of the biochar are very important points.
Okay.
Okay, thank you so much for
Thank you.
We are ready to go.
Okay. Good morning, everyone. I'm Nicola Cornero, a PhD student in Sustainable Development and Climate Change at the University Institute of Pavia.
Sir, just a minute.
Sorry?
Yeah, so we will introduce a speaker for our audience. The next and the last speaker is Mr. Niccolò Zornero from University of Parma, Italy. And the subject is enhancing urban well-being and resilience through nature-based solution, a new project on blue-green spaces and multifunctional reforestation. Thank you. Yes, please, you can start.
Okay.
Hello? Yes, sir,
hello.
you are ready to go.
Okay, perfect. Good morning, everyone. So I'm Niccolò Cornero, a PhD student in Sustainable Development and Climate Change at the University Institute of Pavia. And this is a national PhD program. And my host university is the University of Parma, where I conduct my research with Professor Elena Maestri and Professor Nelson Marmiroli. In this occasion, I will talk to you about the BEPS project. This is a project founded by the Ministry of Health and follow with the the National Complementary Plan for the Investment, and it follow the E.1 line about healthy environment, biodiversity, and climate. This project also is aligned with the United Nations 2030 agenda, and it follows several objectives. But what does WEB stand for? WEBs means the good use of green and blue spaces for the promotion of health and well-being. The aim of this project is to promote the use of blue-green spaces as multifunctional spaces, so make them accessible and safe, capable of stimulating participation, increasing the community cohesion and generating health benefit. So we can say that the link with the ecosystem services is direct. With this QR code, you can view the website that is updated with all the news. Now let's talk about the BEPS number. The regions involved are 4 in all the Italy, and the project covered about the 22% about the national territory. There are 3 national agencies involved and also 3 universities. In this project, we have 3 main actions planned. The first one is the collection and production of scientific evidence on a local, regional, and national level. The second one is to implement a multi-level training with the practitioner, public administration, student. And the third one is focused on the communication to stakeholders, including citizens, and the production of information material as the final output. In this slide, we can see all the operative units involved in this project. In this occasion, we focus our attention on the specific objective number 3 of the project. This objective has 3 aims. The first one is to study the multifunctional reforestation and its impact on earth, environment, biodiversity, and climate. The second one is the analysis of existing experience in the field of study to obtain indication on ecosystem services and design multifunctional green and blue spaces. And the last one is the realization of an atlas of plant species and guidelines to bring decision makers to reforestation based on a scientific evidence. Linked to this 3 actions, there are some indicators that I will show you in the next slide. The indicator number 1, linked to the first action, is the critical analysis of the current approach to reforestation. Multifunctional reforestation is a land management practice that aims to restore or increase the forest cover of an area, reach several objectives like the ecological, economic, and social. In contrast to traditional reforestation, the multifunctional reforestation views the forest as a complex ecosystem capable of providing multiple ecosystem services. In this occasion, we created a database with all the projects in Italy. Some projects are at the local level, and other one is on a national level. One of the most important projects near our university and in our region is the Chilometro Verde Parma. After this, we created a database and we analyzed all the project, and we saw if in the project talk about ecosystem services, if the project is about an urban or a rural area, if there are reports online, guidelines, and also if the project is funded with public or private money. The second indicator linked to the second action of the objective is the study of ecosystem services. In this project, the first action was to view the definition of ecosystem services in the scientific literature. We started to analyze the international classification of ecosystem services, for example, the MEA, the Millennium Ecosystem Assessment, or the CICES, the Common International Classification of Ecosystem Services. After a review of the literature, we found a lot of terms referred to ecosystem services, also with a lot of different definition. So we created a glossary about the urban ecosystem services. This is an example of our glossary. Every terms have its definition, and it's also useful because with the filter you can choose the categories and see all the terms related and the definitions. We classified them with the CICES category and the Millennium Ecosystem Assessment categories. Then, after this work, we found and we defined some macro categories about the urban ecosystem services generated by green and blue infrastructure. Started from there are some physics like the removal of pollutants from the air, the water management and the water quality, the erosion control, the biodiversity conservation, and also urban ecosystem services related to recreation and aesthetic benefits. One of the most important urban ecosystem services for our projects is the mental the ecosystem service that provide mental and physical well-being. So green and blue areas provide opportunities for outdoor physical activity, implementing the mental and physical well-being of the citizen. Now, the indicator 3 and 4 talk about the creation of the atlas. These are some facsimile sheets, and every sheet is about a plant species. This is the structure. We have some physical characteristics and other characteristics related to ecosystem services, like the maintenance, the ecosystem services, and also we try to found these services. These are the ecosystem services under study. For example, the power of mitigation of temperature extreme or the urban heat island, the power of shading, the power of noise reduction, the surface runoff reduction, and also the ecosystem services linked to psychophysical benefits. In the first step, we will try to found these ecosystem services in the scientific literature, found case studies or review. We talk about the ecosystem services, and maybe in the next step we will try some methods to calculate them ourselves. This is the process that we use to create the plant atlas. First, we review all the green regulation in provincial capitals, and we found about 100 plant species. Then we define about 4 plant species, the most common, following the comparison of this document. Then we conduct analysis of existing tools, and they were collected, reviewed, and compared. After this step, we created our standard sheet of our atlas, try to improve and implement the existing tools. So, in conclusion, we can say that the BEPS projects promote research, training, and communication initiatives to support the development and proper use of green and blue areas. By providing guidelines, like for the output, the project aims to guide local decision makers in reforestation practice based on scientific evidence. This is all the authors that are collaborating to this project, and thank you for the attention.
My question is, whatever you have presented over here, is it a project kind of thing which you have elaborated, or something that you yourself has done?
I didn't hear you well.
Okay. Hello? Okay, so the question is, whatever you have presented, that is on the solutions for the blue green spaces and multifunctional reforestation. So it's a elaborative project. It seems to be that this is one of the project which has been elaborated over here, various points, how to take care of those things. But is there anything which you have done yourself in these PPT presentations?
I did myself. Sorry, I hear you very bad. I just hear the last part of the question. If you want to try to tip the question in the chat. Yeah, I followed all the process, but yeah, I started to did the plant atlas. So the project regards 4 regions in Italy. So I review all the green guidelines of the capital of the regions, and yeah, I made a list of all the plant species, and then by comparison, we found the 40 species and start to build the atlas, and with also scientific review. So yeah, I did the scientific review about the ecosystem services to build the glossary, and with all the categories and all the terms referred to ecosystem services. And after this work, I I start to build the plant atlas. This is also a cooperative work.
Yeah, okay, thank you very much. Maybe there are some questions from the audience. There is one.
All right, hello. Nice presentation. Thank you very much. My name is Botany from Botany University. So my question is that this, I have seen this kind of project, and always the question, what's your provision to manage conflicts that you will have? Because you have ecosystem, that means wildlife, and the people will clash, and things like that. So is there any provision in the project to manage your conflict of interest because you are a user?
Of the project and the formation. I think it's very important to talk with citizen and try to explain also this project to the person who live in the city and in urban areas. For example, an initiative that we had in the past months and also now is the creation of a glossary of a questionnaire to try to understand how people live these green and blue spaces in the city. Also, the blue spaces are important because maybe a lot of people think just at the green spaces, but yeah, I think that it's important to talk with the citizen and try also to do maybe some case studies on the field. Try also to understand what people what people desire about these spaces in the city. So the project is about to promote the well-being of this space. So I think it's correct to start from the real needed of the citizen of this area. Yes.
This also? Okay.
For the first question, we tried to define these services. So, for example, the damage caused by the roots at the street or the private property of the citizen. Also, when there was big storms in the city, in Italy we had a lot of damage to private property, or maybe the allergy. So a lot of people have this disease, or maybe the animals, or maybe the wild animals in the parks. Or another important these services is the security. For example, a lot of people maybe are afraid to walk in the park during the night. Or another is the problem of maintenance. For example, because it's the maintenance has elevated cost for the municipality, and this is some of the services that we try to study, or maybe also the availability of the plant species on the national territory, maybe because practitioners choose the wrong species that is difficult to find in the national territory. So this is cool. This is another services. And for the second question, for the first step, we studied all the urban ecosystem services and tried to put all together in the atlas. And to evaluate them, our first step is to find a case study or a similar example in the literature, and try to take them in our territory with our common plant species. In the next step, maybe we can improve the evaluation method with our study.
Okay, thank you very much.
Okay, so with this we conclude the session over So the phytomonitoring session is now being concluded. There were three different presentations. One is from Mariam, Missouri State University for enhancement of restoration of mine tailing. And the second one from Petro, very, very nice presentation. And followed by the Nicolo presentation for enhancement of urban well-being and resilience. Thank you so much, everyone.
Good afternoon all. Good afternoon all. Can you please wait for some time? Yeah. So good afternoon to all. We have a presentation from Michael Mensch on can beta cyclodine paid with either compost, biosurfactants, NPK fertilization, earthworms, or bioaugmentation enhance dieldrin phytoextraction by cucurbita pepo as the first presentation. I welcome you.
Thank you,
Please.
Chair. Good afternoon. For those who are not familiar with organochlorine pesticide, most of them are insecticide, especially you have the picture of the Colorado potato beetles. So it is to fight against such pest, and especially most of these compounds are belonging to the dirty dozen, and they are classified as persistent pollutants, organic pollutants. They have been banned mostly in the world, but they are persistent. Do we have evidence that we have pollutant linkage and still exposure of human being to such compound? Yes. If you look about the peristaltic adipose tissue in men and you sample, especially you can see in France for male, you can determine more than 30 organochloride compounds, and especially PPDDE, a derived product from the DTT, so very long time that is been banned. But you can see with a red circle the presence of dieldrin, and we have also exposure, evidence of exposure in the French West Indies in tropical area. And you can see it's even worse regarding the organochlorine compound, and they have more chlordecone and less dieldrin, but they have both. So now, in France, it is banned since the 17th, but we have case studies, especially in the northwest of Bordeaux or in French Brittany, and we have this concentration of dieldrin in the soil, and it's a problem especially because you know the cucurbits, they accumulate in fruit, squash, pumpkins, zucchini. We have evidence that they accumulate such product. It will be emphasized by Gregory Collen in another talk, so I don't extend my talk about that. But now the question is, can we phytomanage such soil to minimize the transfer to food chain, and especially the purpose it is to phyto-extract the dieldrin from the soil. So we have evidence from the field. Really, it's not spike. I don't like to spike the soils. So we are really working with real soil contaminated by long-term application of dieldrin. Especially, you have to take care of the homogeneity of your field, because the distribution of such product is really, they are really split, and it's not homogeneous, so you have to take care, and especially why we are working with large pots that we place. So we sample the soil, we determine exactly the dieldrin in the soil, and we work with a pot, we fill with the soil, refill with the soil, but we place outdoors, so it's like a field experiment, but we know exactly where we have and which concentration of dieldrin we have in the soil where we are working. We were testing different cucurbits and other plant species, and especially we determine here that we have accumulator variety of zucchini and non-accumulator. What I mean accumulator, it means that you have both accumulation in the leaf, stem, and the fruit, and non-accumulator that you have less accumulation, that's the red circle. You have some cultivar with less accumulation in the fruit, but still accumulation in the leaf. So this is really interesting because you can, the farmers, they can produce, but at the same time, they can remove a part of the dieldrin from the soil. So now, so this is the quantity, Greg will demonstrate you, this is the quantity of dieldrin you can remove. Now the question why? How to speed the phytoextraction of the labile dieldrin? Because we don't care about the total dieldrin. What is very important, it is the labile dieldrin that can be uptaken by the root. Which zucchini variety, and especially we were testing accumulator in the fruit and non-accumulator in the fruit to determine what could be interesting. And now one of the hypotheses is how we can increase the bioavailability of dieldrin so that to maximize the root uptake. And especially we know from the literature that we have a product like in the family of dextrin, especially the beta-cyclodextrin, because it costs nothing, it's very commercial, availability is high, and the price is very low. And I will explain what is the effect of cyclodextrin later. Now, we can also use biostimulant to promote the plant growth. Foliar silicone application of silicium foliar fertilization could be interesting. Silicone, it is, you know, dieldrin is lipophilic. So if you have much more wax, much more suberin, maybe we can increase the uptake of dieldrin. Selenium, it is a cofactor in some enzyme like GST, so detoxification could be interesting. We want also to improve the biodegradation of the and increase the labile pool of the dieldrin by degrading the organic matter or dehalogelating the dieldrin. So we were also testing bioaugmentation, so to inoculate some strain, microbial strain into the soil. Supply organic matter to promote the plant growth and promote the microbe. And we can have also a mixed culture with vetiver or alfalfa, because vetiver has a high uptake of dieldrin into the root, and alfalfa is promoting the microbes. I just mentioned now, we have also another treatment with earthworm, bioaugmentation, and compost. Earthworm, the purpose was also to degrade the organic matter and release the dieldrin, and so that we can have much more uptake of dieldrin by the roots. This is beta-cyclodextrin. What is very important, you have a hydrophilic outer layer, and inside you have a cavity that is hydrophobic. So dieldrin can enter into this cavity, and then it could be uptake by the root of zucchini. Now, this is a scheme of the treatment. We have 3 doses of beta-cyclodextrin. The different treatment, microbial bioaugmentation, fertilization with nitrogen, phosphorus, and potassium to increase the growth, compost, foliar silicon supply, foliar selenium supply, mixed culture, and the combination of earthworm, bioaugmentation, and compost. And the purpose was to determine, of course, the growth of the plant, the concentration and phytoextraction of dieldrin by the root and the above growth part. Now, it's just to show you where it is located. It is real soil coming from the field. This is the content of dieldrin, but take attention, we don't have only dieldrin. We have other organochloride, but today it's too much, it's enough with dieldrin. This is the effect on the biomass. It's a pot experiment here. We know exactly the dieldrin concentration in each pot, and we have a decrease when we increase the cyclodextrin, we have a decrease of the biomass. The reason is we increase the hydrophobicity of the soil, so the water uptake was reduced, and in fact the plant growth was reduced. It was not expected according to the literature, but that's what we got as an outcome. It is both cultivar. Now, about the shoot dry wet yield. You see there is no evidence of the treatment except one at the CD 0%, so no supply of beta-cyclodextrin. Then we have a positive effect of the combination of earthworm, bioaugmentation, and compost. This is the only one. All other cases, we have a decrease or no significant effect. So no effect of the treatment, no effect of the beta-cyclodextrin. Now, about the concentration of dieldrin in the leaf or in the stem, when we increase the cyclodextrin, you see the the dieldrin concentration increase in the leaf. It is why? It is because we reduce the biomass. So the dilution effect is counteract, and in fact, you concentrate the dieldrin in less biomass of the leaf. But what is also partly unexpected, it is a decrease in the stem. Decrease in the stem, except one, it is when we have earthworm, then we have still an increase of the dieldrin concentration. Without cyclodextrin, we have an increase in the stem of the dieldrin. Now, about phytoextraction, that's what is the major outcome we were looking for. We have only, because you know the phytoextraction, it is a combination of the shoot dry wet yield and the concentration of dieldrin. We have only one case where we evidence an increase of the phytoextraction of dieldrin. It is the full combination earthworm, bioaugmentation, compost without cyclodextrin. Some treatment were even decreasing the phytoextraction. Mixed culture with vetiver, application of selenium, decrease a little bit. So take-home message. Influence of cyclodextrin application. No evidence of dieldrin phytoextraction by zucchini. If we increase the cyclodextrin into the soil, we were not increasing the phytoextraction of dieldrin. Shoot dry wet yield was reduced by cyclodextrin application. Without cyclodextrin, dieldrin amount extract was multiplied by 2 when we applied the treatment with combining lombric, compost, and bioaugmentation. It was due to the increase in shoot dry wet yield and stem dieldrin concentration. And we have an increase of foliar dieldrin concentration compared to stem when cyclodextrin is supplied. What are the questions now and the perspective? Earthworm. Do we have plant residue digestion in the GI tract that explain a release of dieldrin and increase, for example, of dieldrin in the stem? That's a question. So we need to determine the dieldrin in the worm casting, and also we need to demonstrate that in a long-term field assessment. About the molecular mechanism driving dieldrin uptake by root, we are working on, and especially we are focusing on some proteins involved in the uptake of dieldrin, and that could explain why the cyclodendrin is not working with zucchini, because apparently the supply of cyclodextrin is competing with this protein released by the root of zucchini. Translocation and storage, also we have to determine the molecular mechanism we are working on. Microbial strength for dehalogenation and biodegradation, that could be another way to degrade and to decrease the uptake of dieldrin in the case if you have a zucchini cultivar accumulating in the fruit. Bioavailable dieldrin striping removes this, our advice is to remove the senescent above ground part, compost them, and after that you can treat by different way. You can use supercritical CO2 to, let's say, degrade the organic matter. You can vitrify and obtain a glass, totally insoluble compound inside. You can also use some fungi to degrade such product, and maybe other technology, but it is your work in the audience to discover how we can treat such plant residue in a better carbon way and a better carbon balance. So it's all for my presentation. If you have questions, I'm open, and I just show you all the people that were working, especially all the students that were working with me and Gregory and Marie-Cécile Alfolder during this work.
Thank you, Michael. So now if you have any questions, then he may answer your questions.
Did you by any chance use sunflowers also? Sunflowers. Sunflowers.
Yes, we were also testing sunflower, and there will be a lecture by my colleague Gregory Cohen about sunflower.
Oh,
yes.
thank you. Look forward to it.
So I don't want to pitch his lecture. So he will answer, he will give you data about sunflower.
Nice presentation, Michel. So a quick question. I mean, I have actually many questions, but so at the beginning of the presentation, you say that different varieties have different bioaccumulation rates. Is there any information about what caused this differential bioaccumulation?
Apparently, it's due to different pattern, different expression of gene coding for some proteins. But Gregory will talk about that. We are in the future. We have a collaboration with Japanese colleague about such molecular mechanism. Yes, the purpose is to use all the omics technology and testing the different cultivar of zucchini, especially zucchini, and using the genomic, transcriptomic, and proteomic to determine why we have such different behavior, and especially when you have the loading from the leaf or from the stem, I mean, or maybe from the xylem into the fruit. That's the key, that's the frontier of the science at the moment, and that we want to do really the next 2 forthcoming years. Yeah.
How interesting.
Any other questions? Yeah, maybe I will ask you some. So, did you identify in the literature any dieldrin degrading microorganisms?
Microorganisms. In fact, I have determined the microbial strain. I cannot show you today, but we have determined in this soil the bacteria strain and the fungi. And especially you have some strain interesting because they can drive the dehalogenation of chlorine in this molecule or the other organochloride compound. Some of the fungi, they are also able to produce catalase and also enzyme, but to include oxygen into the molecule after you remove the chlorine. And that could be a way also to speed up the biodegradation. But one of the key also it is to avoid that you restitute, that you resupply the plant residue. We have to decrease the fresh plant residue into the soil to avoid that you resupply the dieldrin into the labile organic matter pool. But that's true. We have determined the microbial strain, yes.
That's very nice. Any other questions? Yeah, if there are no questions, I thank Professor Michael for his wonderful presentation. Let us go quickly to the next presentation. Yeah, this one. Sure. Yes. Yes.
So next one is a recorded session.
Recorded session.
Okay. You hear me? Yeah, the next one is Dr. Douglas Birch from Ronball in the US, and it is installation, care, and maintenance of phytoremediation plot at a chlorinate volatile organic compound brownfield site, and he explains the lesson learned. So you have the floor, Douglas.
Hello, this is Doug Burge. I'm a geologist with Ramboll Corporation in the United States, based out of St. Louis, Missouri. And I wanted to just give a quick talk about a site I worked at and did phytoremediation at, and some of the lessons learned from our implementation of that. Quick outline of my talk. I'm going to give a quick talk about the site itself and the background. Then I'll jump into the lessons learned from planning and preparation of the site, the planting, and the maintenance. A very important takeaway is that to be successful, phytoremediation requires upfront planning and also long-term installation and care. We were lucky enough at our site to have a local site caretaker who was able to keep an eye on the trees on a semi-frequent basis. which was very helpful. You just be aware that phytoremediation is not a set it and forget it kind of a remediation process. You need to maintain those trees for them to be successful. This site we're talking about is a former manufacturing facility. It used degreasing volatile organic compound chemicals for 35 years. The facility was closed in 2008, and ultimately the client made the decision to demolish the facility in 2016. They did leave in some of the hardtop pavement, concrete and asphalt, but all of the buildings were removed. At this site, groundwater is approximately 2 to 6 feet below ground surface. And below that, at about 15 to 20 feet below ground surface, is a very tight clay till aquiclude that keeps contamination from flowing downward. So ultimately what we have is a thin seam of impacted groundwater from about 2 to 20 feet below ground surface, which kind of made it an ideal situation for the implementation of phytoremediation. Groundwater at this site, unfortunately, flows to the south, and I say unfortunately because you can see to the south is an adjacent residential area, so that created its own set of issues. There's 4 primary source areas of impact on this site from the former manufacturing, and 2 plumes emanating from that. Total VOCs range up to approximately 81 parts per million before we did any of the phytoremediation. And there actually was some encroachment of this contamination onto adjacent downgradient properties, which was definitely a significant issue. Ultimately, this site was enrolled in the Illinois Environmental Protection Agency's voluntary cleanup program, and we were able to complete a successful risk-based closure of the site. And after the site was closed, our client was able to sell the property to the local city government, and the plan is to eventually redevelop this for future uses. There's 12 individual tree plots on the site. We did planting in 2018 and also planting in 2019, 2 separate planting events. So we partnered with Intrinsics Environmental on this site. They not only provided the bacterial endophytes for inoculating the trees, but they also provided some upfront planting and some hands to help with the planting. In total, we put 600 primarily hybrid poplar trees in the site, some willow trees, and we cover approximately 1 acre of the site with the trees. And they were put in a focused fashion to interrupt the groundwater flow, capture, treat the groundwater that was impacted, and also to treat our source areas. So as I mentioned, the facility was demolished prior to implementation of the phytoremediation, but there were still some active on-site utilities. So private and public utilities are obviously something you need to be sensitive to when doing any kind of subsurface work. There actually were overhead and buried utilities we had to be aware of. There were overhead electric lines. I've highlighted in red where a lot of these utilities ran. Along the main street out front of the former facility, there were electric lines running on either side of the street. Also across the full site and around the street were sanitary sewers and storm sewers. There's also buried water lines and buried communication lines. So when you're clearing a site or getting prepared to do any subsurface work, you need to make sure you're not going to interrupt or damage any private or public utilities, buried utilities. So in the United States, we have public utility marking entities that will come out and mark the public portion of the utility lines. Then we will normally have a private utility marker come out to use geophysical techniques and other instrumentation to mark the private utilities to the best of their ability. We also look at historic site drawings. When we're at the site, you look at site conditions to try to further find lines of evidence of where these utilities run. You just want to avoid them. Ultimately, if you're not sure where things are, you can also do soft dig, which involves using like hand augers or air knifing to clear utilities so you're not drilling with mechanical means and damaging anything. The site maintained the concrete floor of the former facility, or a good portion of it. Here's a photograph of the floor of the facility. You can see in the foreground there's actually a flush mount monitoring well in there that we installed. But to install our trees, we not only had to clear it for utilities, but we had to cut through the concrete, saw cut it, break out the concrete, and then drill down through that section there. So we had to plan ahead and in advance of any planting, mark these out and have everything opened up. Similarly, up front of the facility, they maintained some of the asphalt parking lot. Now the parking lot was much easier to drill through, but unfortunately we did discover that there were actually several layers of asphalt in front of this facility when we started drilling, made it difficult to drill through with the augers. They were mounted on these Bobcat machines. So on the right you can see we actually saw cut the concrete slightly, or the asphalt, in advance of the drilling to help make it easier for them to drill through. Just something you just had to kind of be aware of. If it had been thin asphalt, we probably could have gotten through it relatively easily. It ended up being kind of thick, but we were able to work around that problem. When we planted the trees, it was very early spring, and we had one day we had very cold temperatures. It was in March or something like that, and there was a snowstorm came through and a very cold snap of an evening. So you can see the trees up here on the right is how they were delivered. They were just 10-foot poplar poles in plastic. However, they had predicted some very freezing cold temperatures one evening, so I went out ahead of time and got some moving blankets, covered them up and tucked them in for the evening just to help them not freeze before we were able to plant. Fortunately, it was just a single day that we had the cold temperatures and things warmed up thereafter, but that was just something that we kind of ran into, unexpected, but something to be prepared for. Here's some pictures of our planting itself. You can see the inoculation of the trees ongoing on the left side. And we're putting the tree poles into the ground on the top photograph. Through asphalt and in the grassy area, we're also using tree protector tubes around the trees because of the potential for deer. It's a semi-rural area where this is. So we wanted to protect the trees, but we had kind of stair-stepped and logistically done this. We had gone in ahead and done all of the boring and prep work and had everything prepared. And then we came in, inoculated the trees, and then we came in and planted the trees. So it's kind of a stair-step thing to put in. You can't necessarily do everything all at once. You have to logistically do it in stages. And you can see here within 6 months we had some very significant growth. Things grew quite well there. However, we also had some other issues. Winds and storms can create issues. And shortly after planting, we had some very strong straight line winds. And our site caretaker visited the site very shortly after and discovered that some of those protector tubes had actually folded over in the wind and bent over the trees with them. So it's not a pretty picture here. We were lucky that it did not actually snap or damage the trees, and that our site caretaker was there in a timely fashion, was able to upright these, and the trees all survived. Our site caretaker put in some additional tree stakes to hold these up, so that did not reoccur. But just again, that's a nice lesson learned. You know, significant weather events can have issues with the trees. So you need to make them survivable for that. Drainage was an issue at this site. As I said, we were planting onto the existing concrete floor of the facility. Well, that floor was not made, it had a roof over it. It was not made to drain itself. So you can see here up on the right, there were some areas where water ponded. It wasn't necessarily, it wasn't feet deep, but it was enough that it could create some potential issues with the trees. So before we were planting, we realized where this was going on, and we worked with our site caretaker and some local contractors, and we utilized existing trenches that were in the concrete floor, and also utilized existing storm drains that were in the floor to capture this water and make it go away so that we didn't have water ponding in these locations. Worked quite well, but something just that was a planning and preparation phase. Abrasion was an issue we discovered in year one. Those tree protector tubes that we use, those are the white ones, they had a hard end. So what ended up happening, we discovered, was when the trees started blowing in the wind, they started rubbing against that edge of the protector tube, and that unfortunately created some abrasion of the bark, and that is not a good thing for trees. The bark is there to protect the trees. That allows canker, insects, other things to get into the trees. So during our second round of planting, we still maintained the protector tubes, but we were able to source some protector tubes that had a fluted or rounded top, which kind of diminished that abrasion and the rubbing. So that's something I would strongly suggest when you're planting the trees. Either keep them from abrading and rubbing, or put in something that's going to minimize that potential damage. Some other abrasion we had was from the deer. The deer didn't really bother our trees very much. It's a rural area. We were near an active street, so the deer tended to not come out to the trees. But later on in the planting, we had removed a lot of the tubes, protector tubes, and we discovered that some of the deer had come over and rubbed on some of the trees. And on the right, you can see a picture of one of the trees, some of the damage that was incurred. Now, it turns out it was only a few trees that suffered this damage, and they were on the very edge, so it wasn't very widespread. And actually, this tree survived and did quite well, but that was the reason we went with the tree tubes, to try to protect them from the deer. In addition to abrasion stress, there's chemical stress. Our site had, I said, like up to about 81 plus parts per million of VOCs. Well, we use endophytes to help protect the trees from that and make them more survivable. The photograph on the left is of one of the trees planted where there's over 80 parts per million of VOCs that it's capturing and treating. You can see on the right are some trees that are planted in an area that have much lower VOC concentrations. And you can see the difference in the size of the leaves. So the trees were undergoing some very stressful conditions with the very high VOC concentrations. Now the takeaway is that the endophytes, I think were it not for the endophytes, these trees may not have survived in the very high contaminated areas. So they may have been stressed, but they survived, and they used groundwater, and they treated groundwater. So that's just an important takeaway, and just kind of a nice comparison of what we were able to accomplish using the endophytes. Another issue we noted out there, which was a bit of a surprise, was the drought stress. Our site caretaker came out to the site and gave me a call, and the trees are turning yellow. Well, kind of dug into it more. Well, it was became a very dry period of year, and the trees were just automatically or naturally shedding some leaves so that they could survive the drought conditions. And it kind of looks alarming at first when you look at these, but they all survived. but they were just backing down their production because they didn't have as much water to, they had to kind of retract to keep themselves surviving. So just be aware, I guess, of the different signs of stress that you're looking for at this site. At the site, we had some phenomenal growth. You can see here's just the 3-year mark of planting, from planting to 3 years. 2 views before and after. So we had some phenomenal growth of these trees, and they used a lot of water and ultimately led to our successful closure of the site. Just a quick snapshot of what we were able to accomplish in the source areas and some of the hot spots after we did some pre-planting sampling of the groundwater, and then as time went on, we did additional groundwater sampling. And at the time of closure, since we had started, we had some significant reductions in the VOCs measured in the groundwater, up to a 97% decrease in concentrations, which allowed us in relatively short fashion to close the site. The trees did a phenomenal job. I couldn't be happier with what we were able to accomplish out there. Another added benefit, lesson learned, something the client really enjoyed or was appreciated, as well as some of the regulators, was the carbon sequestration that the trees provided for. I did some back of the envelope calculations for trees and how much carbon dioxide they were removing and other benefits using the U.S. Forest Service iTree software. But these are for single trees, so a single tree in a year is removing about 68 pounds of CO2. Well, you multiply that by 600 trees that we have there. So it starts to be some very significant numbers of carbon being sequestered, and that's a very big thing to make sure this is a very green technology, and it's just another added benefit of using phytoremediation. Ultimately, we had fast-track closure on this site. I mean, within 3 to 4 years of our initial planting, we achieved our remedial objectives and closed the site and sold it. It was a very successful project. Very proud of this project. And, using the phytoremediation, we had great acceptance by the client. The Illinois EPA was on board with this. The local city government was on board with this, and even the local residents, who were obviously concerned about the contamination, were pretty excited by the phytoremediation and work we were able to accomplish. So in 2023, we actually finally closed the site. I decorated a cake out at the site for a final walkthrough. So here's my little happy closure cake. And with that, I thank you, and please let me know if you have any questions. Thank you.
Okay, thank you. We will give you some questions.
Thank you for a very nice presentation. 2 questions I've got. One is, what was the spacing?
Yes, it's good joke. Well done. In fact, by chance, I'm doing exactly a similar experiment in France with poplar. And one of the questions we would like to ask is if we measure chlorine into the leaf to demonstrate that we have TCE accumulation or probably degradation due to the endophyte or due to the enzyme in the poplar leaf. But I missed to do it. Maybe he did it. We will see. What will be your question?
My question is, on which basis did you design the spacing between the trees,
and the species?
I can give you my knowledge about the plant species and especially poplar. In fact, the information comes from my friend, Professor Jaco van Gronsveld in Hasselt, and we were working with the Ford company 25 years ago, and he discovered the combination of the assemblage of poplar and endophyte and mycorrhiza to decrease the TCE concentration in the plume below the former facility of this Ford site. And most of the information was coming from Jaco van Gronsveld, working with Niels van der Lully. Niels van der Lully was working on the endophyte. And another part of the information was coming from the state and from a man named Louis Litch. He was working with poplar, and we know that poplar is very, very flexible, and it can survive drought, salt, many things. He is not looking only for water, but he's pumping for water, but also resistant to drought. So, and I'm testing many trees at such kind of site, and especially willow is less efficient than poplar. Miscanthus is efficient, vetiver is efficient, aster are efficient, but the one producing more biomass, it is poplar.
Yeah, what you say, Michel, is entirely correct. It's very fast-growing species. Okay, I mean, there are different poplar species, but they grow very fast. So it's really the plant of choice for phytoremediation, you know, in a temperate area. So I think they are used in many places. Yeah. Sorry, we have to move to the next lecture. You can chat together. But it was very interesting.
Yeah, we have the next from Azam Noori. She will be presenting on assessing phytotoxical impacts of PFOA on Lemna minor. Azam Noori, are you ready?
I'm ready. Hi.
Okay, start.
Can you hear me now?
We can.
Oh, awesome. Perfect. Too many stuff here. All right. Hello, everybody. Good afternoon. And thank you for coming to this session while you could be somewhere else or enjoy the weather outside. My presentation is about PFAS, particularly PFOA, which is a type of PFAS. A lot of you know already what is the content PFAS. Some of you, especially younger audience, are graduate or undergraduate student, may not know. So I give you a little background on that. So the result I'm presenting, I'm going to talk about the research I did as a pilot. So it is a pilot study providing some information. So what are PFAS? PFAS, this name, this abbreviation, stands for per and polyphenol substances, meaning there is a strong bonding between carbon and fluorine. That makes this group of chemicals very resistant to temperature for any chemical degradation. So they got the nickname, yes, it doesn't work. It got the nickname of forever chemicals. Then we have their application. So if you consider chemicals being resistant to temperature, resistant to grease, water, so then therefore they have a lot of applications, which I will talk about them in few, some of the examples. And this widespread application results in the release of them everywhere. So you find PFAS in air, in water, in soil. So everywhere you go, they are available. So they got the nickname everywhere chemicals. So you search everywhere chemicals, forever chemicals, that's what will pop up. So applications. These are some of the examples of the chemicals do have PFAS that you encounter maybe in daily basis. So you don't encounter firefighting foams, hopefully not, but you are using non-stick cookware. If you do, I recommend go home and discard that. Non-stick cookware, waterproof makeups, food wrapping items. Is this okay? Something is off. Yeah. Food wrapping items, these are the things we are using on a daily basis, and they do contain PFAS, a lot of them. A little also history. PFAS were produced in 1940s. They have been released into the market and environment for all these applications they have. In fire fighting foams, they have been used significantly, resulting a lot of contamination all around. So there's a significant issue related to them, the widespread application of them. The examples of the industrial sites containing them or using them as described here. And by the way, the source to all of this, these are interactive maps. You can search for this environmental working group agency. They have the data, and if you click on each dot, you have all the information, the date and the site, all the information are public accessible. So if you look at these examples, for instance, suspected user of PFAS are all those orange color ones. The red ones are showing known PFAS, and it's widespread everywhere. The purple one are the landfill, and which we have everywhere. You can see all across the country. But this is about US. It doesn't mean the rest of the world does not have. It means there are a lot of work being done to collect information. We always have this. You look at health-related cases. We have a lot of data from the societies that they collect the information. It doesn't mean the rest of the world does not have it. Same goes with Europe. So we have this one from the Le Monde website that's also interactive. The keys are there. Known contamination are red ones. All the red sides are showing the level of PFAS release in the environment. PFAS users are in purple color, presumptive contamination. Sites very remote. You can see Iceland. Also, all these areas do have PFAS. And then manufacturers are in dark black color, diamond color. Several of them in Europe, Western Europe. To know how the data appear, if you click on one of them, this one is in Netherlands, shows you 500 nanogram per kilogram. How big is that, or small? What does it mean to have 500 nanogram per kilogram? It's extremely high. It's very high. I'll show you the limit in few. So talking about this widespread release in the environment, you may ask, okay, how is that? Are organisms exposed to it? And the answer is yes. This study here shows over 300 species. Again, this one is from Environmental Working Group. 300 species were detected having PFAS. And that goes from species in environment that are around urban societies or very remote environments, like polar bears or eagles. These are all color-coded for you. Small mammals, large mammals, so across the board, which is like scary. And then you may ask if this is so significant, and we are talking about over 60 years, so 1949 is almost 80, more than 80 years, releasing the environment. How about us? So PFAS bioaccumulation in humans, the data we have, the results we have, again, public accessible. You can find on CDC website. Over 98% of individuals tested were positive. They had detectable level of PFAS in their serum, which is concerning. Now you can ask, well, if that's so significant, why are we all alive? If we are, most of us, so not scaring you, if I do have it, why am I still alive if that's so concerning? The answer is the silent, it's chronic. And you answer this one with the symptoms I'm saying. My mom used to say, rest in peace, she used to say, they release something in our water. And I can see that now. The gradual things we see around us. Weakened immunity, do we see that? Do we see people are not responsive to their vaccines, including COVID vaccines? Do we see thyroidism? Do we see issues with metabolism, obesity, or the other end, and the level of T3, T4 being impacted? Cardiovascular disease associated with high cholesterol. A lot of people saying, I'm being mindful about my diet. I don't know from where I got high level of cholesterol. So maybe look to see what is in the materials you are consuming. Higher risk of particular cancer, that goes to kidney cancer, liver, and pancreas, more significant than others. And the one that's studied quite a lot goes to lower fertility rate and also hypertension for pregnant women and also embryogenesis. The cell division, the pilot studies in the cell cultures in vitro, they have published that they are impact on cellular division. So with all of this considered, you can guess PFAS are in the news. A lot of agencies talk about PFAS, and a lot of news are released every year. So I have examples from Guardian. It goes from new studies, finding something new, saying, yep, it impacts sleep, it impacts the rate of cancer, or so on, goes to cost associated with cleaning, goes with lawsuits. There was a recent one published in New York Times related to some issues with the agricultural field and the lawsuit that comes after that. And then the regulations that people are asking, please regulate this. And then, yes, EPA has been working on this for a number of years. And during this administration, Biden, so I'm talking about the state, they went one step ahead and try to regulate PFAS a little more. So where we were before this, a few years ago, PFOA and PFOS, two key ones, were banned in the state, not being allowed to use them in manufacturing products, especially food items. That's the right thing, there's the food items, so meaning not for food wrapping. But does that mean it doesn't still exist there? No, actually it does exist. And one is PFAS are called forever chemicals. They do not break down easily. Their lifespan is 3 to 4 years. And when you have high concentration, it takes them forever. And also materials coming from other side of the world. They all have. And then it goes everywhere. So here we have the work EPA did. This is also on the EPA website. There are two key phrases here. There's one, maximum contaminant levels. There is another one, maximum contaminant level goals. So if you look at two columns here, the maximum contaminant level for PFAS, PFOA and PFOS, these two I mentioned that basically are banned and very concerning because they impact at the nucleus level up to organismal level. These two are at 4 ppt, so the goal is reduce the level of them to 4 ppt in drinking water, which I'll show you some, maps in few. The goal is go to 0, so we are not 0 yet, obviously. The others are also the PFAS of concern. So here I want also to bring to your attention, when we say PFAS, that phrase is substances. There's another S at the end. There are 4,700 different types of PFAS. Again, these are emerging compounds. They are man-made. They're unsynthesized. They don't exist in the environment. We made them, we released them. For many applications they had for us. So moving forward, in water, how much are they in water? This example we can find on the website from Environmental Working Group. The light dots are showing, light blue ones, they are showing higher than the limit level of PFAS in drinking water. So directly going into people's water. And they cook, they take shower, and they do everything else with it. Then we have the darker blue one that are lower than the limit, but remember they can be accumulated in our body, so we don't want to be exposed to them. The military size, if you are curious, I'll let you go look at that. The military size contamination, the numbers are extremely high. So remember what we saw? 4 ppt was the goal. We want to get to 4 ppt as the limit. The military size, you're talking about 4 digits, 5 digits. So they are very high because of the materials they have used for firefighting and so on. We have some other sites as well. So examples of this site, my college is located here in Massachusetts. So I'll show you next slide here. North of Boston, that's where we are. And you can see in the area that you have higher population when it is dense and you have industrial sites, the contamination is higher. If you click in one of them randomly, I'll just select with that site there, this is the type of data you will see. And PFOA, PFOS are on the list and other concerning PFAS as well. The number you see here, PFOA 11. This is actually less than typical contamination. This is in one of the nicer neighborhoods, Newburyport, and you don't expect actually to have much contamination there, but there is still higher than the limit. And this is the PFOA one. All right, so now going to the study I have. So when we talk about the contamination in water, then you wonder, okay, how can we deal with this? And there are sites that they are using well water. They are still even in the States, so we do have New Hampshire, they have well water. Anyways, contamination in water is a circle, impacts everybody. I look at duckweed, Lemna minor is duckweed, the tiniest plant. The size of the plant is as big as the tip of your pen, so it's very small, has its own advantage for this kind of a study. It's aquatic. It grows fast. So rapid growth reproduction is one advantage for us. It is used for phytotoxicological studies. That's what I wanted to do, to get some idea. So before we go with higher plants, going with plants that they may take months to get the results, let's go with a plant that can grow fast and then give us some valuable data. And it's also used quite a lot for phytoremediation. If you look for heavy metals, it's used for nanoparticles, organic contaminants, duckweed is used. Here you can see the size, how small it is. Here is, these are both same place in New York, I took the picture. From distance, when you stand, it's covering the whole area. There are species of duckweed that are actually invasive. So if you ever want to go and study that, is that my timing? Yep, oh my God, okay. If you want to go and study, be careful that some of them are invasive. All right, so we go fast. In this study, I look at the phytotoxicology responses, interaction with essential elements, and then if duckweed can uptake these materials. Here is the illustration. My student Erin created that. She's an artist and scientist. If you are interested to see more of this work, she is presenting later this afternoon. So here is the schematic view for you, what we did, going from phytotoxicological responses to essential elements and so on. So to do the contaminant exposure and the timing, we did IC50. That's typical study you would do with cancer cells. We did it here as well, so you can do that with your seeds, to find out what concentration of the contaminants result in a 50% reduction of the growth or 50% chlorosis. What is the factor you are looking at? So we did in this study, and that was 0.1 microgram per liter. As far as the timing, we did the same thing, chlorosis assay, and then 7 days. Remember, duckweed after 40 days is not growing well. So that was the max we wanted to look. As far as phytotoxicological responses for photosynthetic, oxidative stress, antioxidative, no significant differences. I put for you just a few examples of antioxidative responses. Malondialdehyde, hydrogen peroxide, anthocyanin. Actually, it's not bad, right? So no significant responses they were able to tolerate. Looking at stomata, so these are immunofluorescence imaging from using confocal microscope. Opening of a stomata is important when you are working with all the species in this kind of treatment, but particularly with aquatic species, you want to know if exposure to a chemical that's acidic and has a lipophilic characteristic impacts the opening of a stomata. And the result was actually yes. was significant reduction of the size of the stomata. So meaning water transpiration is low. We did also looking at cells. So the size of the cells, how cells were impacted. We do immunofluorescence imaging for this too. So you tag the cells with the dye and use the confocal to see them. Similarly, you see the size of the cells, even though visually you may not see that, but when you do the detection and labeling, you see the size of the cell are significantly reduced compared to the control group. Nucleus. That was very important, alarming to me, reading impact on human cells, that exposure to PFAS, in this case PFOA, results in higher, so I have the percentage for you, 27% and 30% increase in nucleus diameter and area. Why is that alarming? Because we are talking about mutation. And visual observation, you can see here, we had more cells being at the metaphase stage in the exposed group compared to control groups. And we have here day 0 and day 7 control compared to day 7 exposed ones. Elemental analysis, we did a set of elemental analysis. The most significant one was with potassium. Sodium I studied, that was also impacted.
You'll be fast.
Sure. Done. This impact on potassium was the highest one, which correlates with stomata opening and closure. My last final slide here is the bioaccumulation. The number, how much was bioaccumulated with duckweed was 9, near 10, so 9.58 nanogram per gram in or on, because it's an aquatic species, and lemna minor, or duckweed. And as far as what percentage is that, the bioaccumulation factor would be over 50%. It was 56. So overall, the toxicological responses was minimum. As far as interaction with the essential element, the most significant one was potassium correlates with the impact on turgor pressure and the opening and closure of stomata. Having less potassium, we had narrower stomata, which has its own consequences for photosynthesis. And that's all it is. So, yeah, I should appreciate my students who helped me. And the PFAS analysis was done by Dr. Jason's, everybody knows him, Dr. Jason's White's laboratory. They did the PFAS analysis. This should be done with certified laboratory, and that's what we did.
Thank you.
Sure, thank you.
Yeah. Maybe we'll have quick 1 or 2 questions. 1 or 2 questions quickly.
Hi, nice presentation.
Sure,
thank you. It's an interesting plant. So you mentioned a series of pharmaceutical effects that PFAS have on the plant. Any indication of the toxicity mechanism, at the molecular level?
At the molecular level, there are not tons of studies that I'm aware of. There are a lot of people are working, but when you look at this, I didn't do myself, and when you look at the literature review, when people are saying potential impacts, the impact are mentioned as potential. So as far as I am aware, it is not the well mechanism known how PFAS are impacting plants. So it's a work to go to find out.
Yeah, very nice presentation, by the way. Thank you very much. Is this PF, I'm talking drinking water sources, if it does get washed into the drinking water source, how can we remediate or remove it, PFs?
Very good question. If PFAS is on the drinking water, how can we remove that? You want to try to not use the drinking water from the sources contaminated. So a lot of towns in where I showed you, they are receiving bottled water. It has its own problem with microplastic, but they receive bottled water that doesn't have that contamination. They are filtered. They are filtered you can use to filter your water.
Maybe I'll, okay, one more. Yeah, maybe you can.
Short. Okay, fine. Very good afternoon. Asam Nuri. I have done a very fantastic
Sure. Thank you.
You asked the
work.
question.
And, okay.
You asked the question.
And thought-provoking presentation. My doubt is that classifying the different classes affected with this PFI, and that fish you mentioned, different fish, US EPA fish, like that one list you shown. No? Fish and US EPA.
The slide with the US
EPA.
Okay.
Okay.
That was a particular one was related to the study was done on the fish that people consume, so therefore it came from EPA. That was different than the fish that not people may eat. So it's something that goes to the food chain versus that versus the marine organism fish that you may just catch in water and study. So that was the comparison, just connecting the dot that US EPA study was about food chain.
Okay,
Thank you.
I'll ask quickly two questions, small, very small ones.
Sure.
The first one is, in the last slide, you showed accumulation of PFOA.
Yes.
But in your previous slides, you don't have accumulation of ROS. How, H2O2, you don't have any accumulation.
Which one?
In your last slide, you have shown accumulation of PFOA in lemna.
That last slide.
Last one. Yeah, here.
Yes.
So PFOA is accumulated.
Right.
But that is not increasing any ROS in the cells. What is it doing inside?
ROS, you are talking about oxidative stress.
Oxidative stress, yeah.
So that goes to our response here. Thank you for this question. When you study ROS, reactive oxygen species is a stress response. So if plants are stressed under these contaminants, they are releasing more of hydrogen peroxide, radical oxygen, or so on. If they are able to cope with that stress, they are releasing antioxidant to bring that down. So our study is showing they survive. We did some chlorosis here and there, but overall, it was not a significant difference when you compare control and experimental. In another way, when I'm saying we propose using these plants for removal, at least this pilot study is telling us, because we didn't see plant dying after exposure. There was accumulation, they didn't die.
Thank you. And the
You're
second
welcome.
one is in your first or second slide, T4, I think it's thyroxine, not tyrosine.
Tyrosine. It was a
thyroxine.
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 Thank you for catching that up. All right, thank you.
Thank you. Now, the next speaker is Benoit van Heeken, and the title is from the John Mason University in the state, and the title is a Safener phenchlorine increase the tolerance of Arabidopsis thaliana to PCB 11.33 p.m. dichlorobiphenyl. You have the floor. No. The Safener increase. Yes. It's a good title. It's a good title. Yes.
Yeah, it's not very visible on the, okay. Hi, good afternoon, everyone. Thank you for coming to this afternoon session. And thank you also to the conference organizers, the Phytotechnology Society and the University of Calicut to invited me here for a talk. All right, so I'm going to talk today about a safener, okay, and PCBs. So I've been working, for many years on PCBs. So you may not know what are plant safeners. I'm going to explain that later. So this is a picture of my research center, which is located in Virginia, south of Washington, D.C. And it's straight on the Potomac, sorry, not the Potomac, the Occoquan River, which is basically going to the Chesapeake Bay. So it's a nicely polluted area. All right. So let's talk quickly about PCBs. You may know about PCBs, you may not, because they are still around. They are forever chemicals, just like PFAS. But the name is actually well chosen because they are still around more than decades after most of their applications have been banned. Okay, so first of all, PCB is a class of compounds. So it's a common mistake. Okay, we talk about PFAS, we talk PCB just like one chemical. No, it's a huge class with different structural feature and different behavior. Okay, so it's a class of 209 congeners with a biphenyl core, okay, and 1 to 10 chlorine atom attached to them. Okay, so about 1.5 million tons have been produced over the 50 years during the heavy industrial production of them. Okay, especially commercial formulation, Arochlor. Very quickly on the background, so they were banned in 1950s because it was people realized that they were bad again for birds and wildlife. Okay, and they were detected during studies to basically detect pesticides. Okay, at that time it was about DDT. Okay, and then this guy emerged. Okay, and so they've been used in multiple applications. I'm not going to go through that, but in capacitors they were used also for all kind of grease, for industrial structures, things like that. Okay, so it's still a major contaminant in many areas. So if you look at the Chesapeake Bay, which is here, Washington is here, the red area there is the one that is contaminated with PCBs. Okay, and there are fishing advisories. No fishing advisories yet on PFAS. Okay, so the concentrations are very significant. My research center is right here. Okay, close to this PCB hotspot. All right, so this one. All right, let's talk a little bit about phytoremediation. So phytoremediation has been recognized a while ago is capable to remove some PCBs from soil and groundwater. Okay, so typically lower chlorinated PCBs are taken up at a higher rate by plants because they are more soluble, less lipophilic, so they go through the plant in an easier way. So they can also be metabolized. That's what interests me more. Okay, so they can be transformed by plants. So this is the green liver model. You may have heard that. So in the 80s, Coleman and Sanderman, there were two pioneers there. They study metabolism of pesticides, herbicides. Actually, in plants, they realized that plants can metabolize them, which was not known. And more than that, the detoxification sequence is very similar to what happened in the liver of mammals. That means a three-phase detoxification sequence that I try to sketch here. So first you have activation, typically by oxidation, at least in the case of PCBs. Hydroxylation, that means the compound actually more toxic, but also more reactive. Okay, and that allows a second phase, which is conjugation, and conjugation to sugar molecules or glutathione will increase solubility and will decrease typically toxicity of the molecule. And in a phase 3, the molecule can be stored in the vacuole or incorporated into cell structure like the cell wall. Okay, in humans or in mammals, you have an excretion phase that replaces this phase 3, but it's very similar. Enzymes involved in different steps here are also very similar. I'm going to talk about that in a minute. All right, so that's just that's the work of Jerry Schnorr, who is a pioneer in phytoremediation. So his group demonstrated multiple times that PCBs could be metabolized. Okay, so typically you produce hydroxylated metabolites. Okay, that's the more visible one. And these are actually very often more toxic than the parent compound. So it may not be desirable if that stops there. Okay, so one of the big problems with phytoremediation of compounds like PCBs, but it's a case actually with chlorinated pesticides we talk about today, and probably PFAS, okay, is that these are accumulated new plants. They can be transformed in more toxic compounds, okay, and then what? Okay, so in the case of PCB, the uptake is very slow. Okay, so and not much is taken up. and then even less is metabolized. So is that significant? Okay, so anyway, there are some need, to boost these processes. So that's why I decided here to sapheners. Sapheners are known for a while. They are actually elicitors of the metabolism of xenobiotics. Xenobiotics, I think everybody understands that, is from the Greek xeno. So they are compounds that are produced that are not natural. They are organic compounds, typically from a human source. Okay, so these these sapheners can boost the metabolism of pesticides, especially herbicides. So they are commonly used commercially together with herbicides to protect the crop plants against the harmful effect of the herbicide. So why is that? Well, simply because these elicitors can boost the metabolism by overexpression of the enzymes that are involved in the transformation of these compounds. So common enzymes include the cytochrome P450 monooxygenase, so CYP. So the CYP will typically perform phase 1 transformation, so hydroxylation. So you have the glutathione transferase or glycosyl transferase that will attach a plant molecule like a glutathione or a sugar to the main, to the contaminant. Okay, so these are very common enzymes involved in this process. So about 20 have been commercialized so far, and they are used for a little while. Actually, something that most people do not know is that herbicides has not been used for a very long time, a few decades actually, but no, agriculture is highly dependent on herbicides, and so these sapheners are commonly used. And if they boost the metabolism of herbicides by producing or overexpressing these enzymes, why not overexpression of the enzymes that can deal with PCBs? So that was just an idea. Okay, so here, the series of these sapheners, you can notice right away that there is no structural similarities between most of them. Okay, you will see that most of them have an heterocycle with a nitrogen, but most organic compounds have, so it doesn't really matter. Okay, so it's a class of contaminants, not contaminants, a class of compounds that are not related structurally, but have similar effects. Okay, that's already a puzzling thing. All right, objective of the study, we would like to determine if sapheners can basically boost the phytoremediation of PCBs by plants. We are going to use as a model PCB11. 3 prime, okay, you number here. I don't know if you are chemists here, but you number the biphenyl here. So 1, 2, 3, 1, 2, 3, so 3 prime, dichlorinated biphenyl or dichlorobiphenyl. Okay, so we are going to test 3 sapheners. Okay, phenchlorim, a related one, which is CMP, okay, 4-chloro-6-methyl-2-phenyl pyrimidine. So here, there is no mistake, okay? You start numbering by the N atom, okay? So it's 1, 2, 3, no, this one. Let me see, I'm going to mess up here. 1, 2, 3, 4, yeah, that's correct. And then if you start here, 1, 2, 3, 4, 5, 6. So it's 4-chloro-6-methyl-2-phenyl pyrimidine. Okay, so that's basically, we are going to go by CMP, which is shorter. All right, so we use Arabidopsis under sterile conditions, growing in hydroponic, semi-solid medium, everything super controlled, because we are going to conduct transcriptomic analysis, so we want to be able to see differences. Okay, so we are far from field conditions, but in cleaner situation, we can see better what happened at the genetic level, so it's a starting point. All right, so a word about this PCB11 is interesting. It's not a legacy PCB. It's still produced today, yeah, because it's a byproduct of the production of some dyes, okay, among other things, like pigment yellow 12. This is this structure. Yeah, I tell you. So this moiety here, is basically a PCB11, okay? So PCB11 is 3 prime, all right? So this has been found in kind of high level in water and air, and especially nowadays, it becomes to be more prevalent because it's continuously produced. Okay, and that's a common mistake. PCBs have been banned, has used for use in consumer product, industrial product as it, okay, but not necessarily as byproduct, okay? So this PCB11 is still produced. So what else can I say? Okay, so let's talk a little bit about what we did in my lab. So we exposed Arabidopsis plants first to this PCB11 to see, if there is any toxic effect. Yes, there is a toxic effect, as we typically see with PCBs. We find an effect concentration 50, which is about 7 milligram per liter. So it's fairly high, okay? So you don't find this concentration in the environment, so that's just for the purpose of lab studies. Okay, so PCB11, however, is much more toxic than all other PCBs that we have tested before. Okay, so it has, the parent PCB has an EC50 of about, I need to speed up, about 7 milligram per liter. So, yeah, I need to speed up. So this is a study that we published a few years back. So the blue circle show basically the toxicity of PCB. No toxicity detected for most of them, except maybe a 4-chloro, but the monochlorinated are more toxic by plants because they are more taken up, and anyway, the level is much higher. These are the 2 hydroxylated PCB you see are much more toxic, okay? So let's move on, up, I'm stuck. I don't know if this is the, yes, that was that. All right, so we tested basically all 3 safeners. Okay, so this diagram shows the control and then addition of PCB3, just PCB3, and then PCB3 plus 3 different safeners. So we have done this experiment twice, which is highly recommended. So what we see is that phenchlorim is the only one that basically changed the toxicity of the PCB, reduce the toxic effect of PCB11 on the plant. Okay, so we are going to focus on phenchlorim and forget about the 2 other ones. All right, so here we test more seriously the effect of the phenchlorim on the toxicity. So these are results based on the biomass of the plant. So we can see the letters indicate statistical significance. So there are about 30 plants per set. So we can see that phenchlorim does not really have detectable toxic effect on its own, but the PCB11 has toxic effect at 30 and at 50 milligram per liter. Okay, and then we see that addition of phenchlorim rescues somehow, it's more visible in this second set, okay, rescue basically the toxicity or reduce the toxicity of the PCB. Well, bingo, that's what we wanted to show. So now, what I typically do is gene expression. So we do transcriptomic analysis, gene expression using either RT-qPCR or we have used microarrays in the past. Now we use RNA sequencing. So we like to see, at the molecular level what's going on. Okay. So for that we have selected GSTs, and this is from a database. Look at the time. Yeah, I need to speed up. So, these. To say that very shortly, these are the GSTs, glutathione transferase and CYP, so the cytochrome P450 that are the most commonly overexpressed by all kinds of chemical contaminants, organic, inorganic, okay? So we have a list like that is based on GeneInvestigator database that doesn't exist anymore, but I have downloaded it. All right, so we use basically a subset of these for RT-qPCR experiment. Well, that's just the heat map that shows, shows that, okay? So here are gene expression level, and surprisingly, so we have tested, RT-qPCR is great because it's very precise and so on, it's kind of affordable, but it's a problem we go gene by gene, okay? So we just tested 10 genes, 5 GST and 5 CYP. Results, well, not much to say, okay? So we see that we have overexpression in most cases, but that we knew already. Okay, we knew that phenchlorim and the PCB they can basically induce these genes. Okay, but we don't see a real differential effect of phenchlorim. Okay, here so I need to explain the graph, but it's shown on the figure. So the blue is just exposure to phenchlorim. Okay, the red is exposure to the PCB11, and the green is exposure to both. Okay. Okay, yeah, I will try to finish. 15 minutes is really short.
Yeah,
All
2
right,
more
so
minutes.
anyway, except this one, we don't see a very striking differential effect of phenchlorim. So it's good that I run out of time, because basically, I have started transcriptomics on the whole genome to learn more than that, but actually the results, I just received them, and well, to tell the truth, I just started to process them, and it's not finished. Okay, I don't have results to present on that yet, so that's actually a good thing, and I have actually more things, so that's typical transcriptomic analysis. You expose the plants, extract RNA, run RNA sequencing. It's fairly easy to do, actually. It becomes more and more affordable. All right, so I move on. I have done other transcriptomic studies. I have a few more things to show about PCR analysis. But in a nutshell, we don't see actually much correlation between the different class of contaminant and the transcriptomic pattern. I think I need to stop. Is that right? Yeah.
You can conclude.
Drink. Okay. So yeah. So it's okay. I have published, all these results, so you can look out. Okay. Before I to go, one question: do plants really sense specific xenobiotic compound, or is just a response, an unspecific response to toxicity? That's a big question, that I have in mind to try to elucidate, but it's a big deal. Well, before that, in mammals, there are xenobiotic receptors. They are known, not in plants. They diverge evolutionary, they diverge before these kind of proteins were developed. So you don't find the homologous genes in mammals. Okay, so in plants, no xenobiotic receptor. So how do plants sense? Either it's unspecific toxic effects, or there are receptors that are still unknown. All right, acknowledgement. My graduate student, Aaron Shafane, that's the plant group, and then I have one student, working more on PFAS, unescapable, funding. Thank you very much. Questions?
Thank you. I think before going for tea, we'll have quick 2 or 3 questions. 2 questions.
Hi, Benoit. Thank you very much. You were complaining that PCR only sees one gene at a time. So are you imagining something multiplex or genomics, transcriptomics, RNA-seq or something?
Yeah, that's what we are doing. That's what we have done, actually. But to be honest, the quality of the RNA was bad, and it comes up with results. I don't know if I will be able to rescue them, or if I have to order a new sequencing. But yes, we use RNA sequencing for now 3-4 years. It's much better than microarrays. I have actually still microarrays rooting in my refrigerator. But no, I use RNA sequencing. And that's what I'm waiting to get clean results. I don't want to present results that are not.
So you expect something more in the cytochrome genes, or?
Well, it will basically go over the I don't know how many genes are encoded there, 23,000, I think. Annotated genes, I think there are 33,000 in Arabidopsis. So yes, I expect to see a clearer pattern, and we have also more tools to look for the enrichment pathway, things like that. So certainly, but good remark. Yes, qPCR is good, but it's kind of limited.
Maybe I'll ask the second one. Before your acknowledgements, we have a last question that you have raised. The question that you have raised in the last slide.
Yes.
Before acknowledgements.
Yes.
So plants do have some specific mechanisms of stress perception or xenobiotic perception. Only thing is, I think we need to find out. Like cold stress responsive genes, they are specific to cold. Heat stress responsive genes, they are specific to heat. Like that, maybe they have something, but we need to find out those receptors which they are present.
Very good remark.
Yes.
Yeah, there are receptors in plants for different kind of effect, like the heat shock or
Cold shock or
different kind of
risk.
stress, but we don't know receptors like in mammals that detect specifically a xenobiotic compound. Okay, so there are no equivalent.
But remember that it is a problem to be too specific. If your toolkit is too specific for detoxification, then you can, if a new stress arrives, then you can be much more affected. So you need to be also flexible. That's why many protein like PR protein are very flexible. So
yeah,
it could be dangerous to be, and you can be damaged and affected by a new stress if you are too much specialized for detoxification. So you need a special receptor, but you need also a very flexible toolkit to adapt to new stress.
Yeah, you two raise exactly the right question. The specificity of this receptor is
Another question before the coffee break?
Sorry.
You are too tired, but it was an excellent session with very good information. So thank you. No. Thank you to all the speakers because I'm sure that you learn very relevant information. So thank you to all. And the strategies, the approach, the tool are very similar for the different organic compounds. How to use, go between control conditions, but go for sampling and real world. How to use the omics now for determination of the molecular mechanism, determining the answer of the organism to the exposure. That's the way, that's the frontier of science. For the scholar, that will be your job.
Yeah, we'll meet again by 3:10. 10 minutes. In 10 minutes, we'll meet again here. It's 3 now. By 3:10, we'll meet again.
Sure, Young Feng. If you don't listen when we ring the bell, I will say it's time. Okay? Let's go. It's a record? Oh, it's a record, so no question. Yes, about the poster. So, sorry. I remember to all the students that you should be standing in front of your poster during the poster session. So, especially the colleague evaluating your poster can ask you some question and evaluate your poster. So please be in front of your poster during the poster session. Now, the presentation is recorded. So just listen, no question.
I'm Yongfeng Xu, and from Institute of Soil Science, Chinese Academy of Science. It is my pleasure to have this opportunity to share our research. My topic is effects of endogenous biohydrogen from a rhizobium-ligand association on microbiome biodegradation of polychlorinated biofenols in contaminated soil. My topic, including the following 3 parts. Firstly, we review the research background. As we know, polychlorinated biphenyls, the abbreviation is PCBs, were firstly synthesized in 1881. They have 1 to 10 chloroatom on the biphenyl. Due to have good chemical properties, no flammability, heat resistance, and other advantages, they were widely used in the electric power industry, plastic processing industry, and chemical industry, especially because of the unreasonable dismantling activity of electrolytic waste. PCBs entered into the multi-environments, including soil, water, and sediments. Therefore, PCBs listed as a kind of priority pollutant. This is the pollution that studies of PCB in the soil. PCB pollution present in our concentration but not air. So, how do we remedy PCB contaminated soil? At present, the main technique for remedying PCB pollution, including physical remediation, chemical remediation, biological remediation, and combined remediation. Among these, bioremediation has a promising strategy due to its higher efficiency, safety, and lower cost. During our research, we found that hydrogen metabolism is widely distributed in various environments, and biological hydrogen as an oxidant for aerobic microorganisms and a reducing agent for anaerobic microorganisms may be the core of microbial community function. In addition, in complex microbial communities, symbiotic interaction between hydrogen-producing and hydrogen-consuming microorganisms may play an important role in pollutant degradation and global carbon cycling. Root nodules of leguminous plants is the largest source of endogenous hydrogen in terrestrial ecosystems. Because symbolic nutrition reactions can annually produce large amounts of hydrogen, hydrogen can be released to surrounding right-field soil and can accumulate to percentage levels from 9,000 to 12,000 as the interface between soil and root nodules. It should be noted that these hydrogen emissions have also been proposed to alter the right field microbial composition, enhance carbon reaction and the degradation of organic compounds, and stimulate plant growth. Based on our previous studies, we found that symbiotic nutrition reactions combining between right field and leguminous plants, reducing degradations and producing hydrogen gas. In other words, endogenous biohydrogen is an obligate byproduct of nutrition reaction process in alpha nodules, which leaks into the reed field. Thus, we have a question. Does the release of hydrogen gas from this system affect the degradation of PCBs by right field microbiota in soil? Thus, we make a scientific hypothesis. We first hypothesized that endogenous hydrogen was selected for contrasting PCB degrading with the related PCB biodegradation pathways, leading to an increase in biodegradation efficiency in the right field. Second, we hypothesized that these contrasting PCB degrading microbiome guide encode different hydrogens and rubisco-lineages known to support hydrogen trophic growth. So, we highlight the results. In this study, we combined DNA stable isotope probing with metagenomics to verify our hypothesis. To achieve this goal, a total of 7 treatments were set up in triplicate. 1. Sterol control treatments. Study is microcosm amended with PCB 77 in sterile soil which will autoclave. 3 times for 1 hour each time at 112.1 degree. 2. 12C Bayer treatments. That is microcosm amended with a labeled PCB 177 anon. 3. 13C Bayer treatments. That is microcosm amended with 13 carbon labeled PCB 77 anon. 4. 12C equals hydrogen treatments. That is microcosm amended with a labeled PCB 77 and treated with hydrogen stimulated the hydrogen emitting root nodules. 5. 13C equals hydrogen treatments. That is microcosm amended with 13 carbon labeled PCB 77 and treated with hydrogen stimulated the hydrogen emitting root nodules. 6. 12C endogenous hydrogen treatments. That is microcosm amended with a labeled PCB 77 and treated with endogenous hydrogen released from mature alfalfa root nodules. 11. 13C endogenous hydrogen treatments. That is microcosm amended with 13 carbon labeled PCB 77 and treated with endogenous hydrogen released from material alfalfa root nodules. Here shows the change in residual PCB and hydrogen oxidation activity in soil in different treatments. PCB biodegradation efficiency increased significantly in soil treated with endogenous hydrogen. Hydrogen oxidation activities was positively correlated with the PCB 77 degradation rate. Thus, the biodegradation of PCB's biosoil could be affected by the right field hydrogen, suggesting that endogenous reservoir hydrogen from rhizobium alfalfa system makes a noteworthy contribution to PCB degradation in the right field. So, according to the current results, a question is who contributed to the degradation. We try to use state isotope probing to identify the active PCB degraders in differently treated soil. Finally, 20 genera were identified as having particular PCB degrading ability. Among these, 2 genera such as Meganetus beryllium and Estumonas were the 2 key PCB degradation tasks shared among all treatment and were stimulated by endogenous with right field hydrogen. All above results confirms our first hypothesis that endogenous hydrogen would favor the development of aerobic PCB degrading bacteria with specific function traits at the genomic level, leading to an increase in biodegradation efficiency in the right field. Metagenomic analysis was used to assess the relative abundance of car group of function genes with a focus on metabolic function of PCB degradation microbiomes. Multiple convoluted pathways for the degradation of PCBs through genomic metabolic pathways analysis were reconstructed. We show here different tasks conducting successive metabolic depth of PCB metabolism. Then we further checked the changes in key metabolic potential of microbiome communities. Results show that right-wheel hydrogen significantly increased the relative abundance of hydrogenous carbon fiction-related amines and biological integration genes in the soil. We also found that the two genera, such as stimulase and magnetovirulum, were effects to contribute to both soil hydrogen oxidation and carbon reaction, indicating this bacteria could grow hydrogen trophically by using electrode deriving from hydrogen for carbon fiction. All these results confirms our second hypothesis that key tax growing hydrogen trophically by using electrodes deriving from hydrogen for carbon fiction were proved to play an important role in promoting the degradation of PCB by endogenous right-field hydrogen. Finally, we proposed a mechanism about endogenous right-field biohydrogen from a rhizobium ligand system driving microbiome biodegradation of PCB in contaminated soil. Namely, active hydrogen trophic PCB degrading microbiomes in the contaminated soil were selected by the right-field hydrogen, which in turn promote the degradation process of pollutants. The last part is the conclusion. 1. PCB77 biodegradation efficiency increased significantly in soil treated with endogenous hydrogen. 2. Endogenous hydrogen select bacteria harboring PCB degradation genes. 3. Different tax conducting successive metabolic steps of PCB metabolism. 4. Enriched hydrogen trophic tax encoding bisphenol oxygen genes driven PCB biodegradation through endogenous hydrogen. 5. Evaluate hydrogen can influence the microbiome ecology and biology chemistry of the ligand right field. Next, the Chinese and future work is that proposed a mechanism for hydrogen production to promote degradation in the symbiotic process. Fermenting a ligand with bio-John remediation technology. At last, I'm very grateful to Professor Yongguan Zhu, Huanyin Yao from Institute of Urban Environment and Professor Chris Greener from Monash University in Australia, and members of Soil and Environmental Remediation Laboratory, Institute of Soil Science, for the assistance in our study. In addition, this research was supported by the National Key Research and Development Program of China, the National Natural Science Fundamental of China, and so on. My presence ends here. Thank you for the attention.
Interesting lecture. One point that we can challenge a little bit is the root of alpha-alpha are not only releasing maybe more hydrogen, but you have all the rhizodeposition, so many other products are released by the root of alpha-alpha, stimulating the answer of the microbes. So I guess it is a little bit much more complicated when you introduce the plant alpha-alpha. Now, to save time, our next speaker is coming from Madras, And from the Indian Institute of Technology, I have my pleasure to welcome you, Samyuka Srinivasan. Sorry, because I'm not used.
Could you please announce about this timing, because the remaining three are competitors. So the timing you have to inform.
Yeah. Okay, we are evaluating the students, so the timing is also important. But in fact, the rationale and the result and the clarity of the slide are even more important than the time. So let's go.
Hanging their poster. Note the time those who have applied for the post
Hello.
presentation
Hello.
requested to.
Are you?
No, someone is
Hello.
in hall. Just
Someone is old.
announcing.
Okay.
Okay. A laser pointer is there? Laser point.
An announcement. To have presentations for poster are requested to the presentation venue for evaluation.
Should not step. Shall I start? Yeah. Good evening, everyone. I thank the organizing committee of IPC 18 for giving me this valuable opportunity to present my research. Today, I will walk you through some of our preliminary research on investigating a stress response of emerging pharmaceutical in plants using genome scale metabolic modeling. So I think you are all wondering why did the pharmaceutical get ended up in plants. So today's, like in this era, we have a drug for every ailment, but less attention is given to the fate of these drugs after use. So after use, these drugs end up in the wastewater treatment plant where they are not completely removed by the conventional systems, and they end up in the treated water also. So when this treated water is used as reclaimed water for irrigation purposes, this exposes the plants to these pharmaceuticals and hence bioaccumulation in the food chain. So, and these pharmaceuticals are known to be biologically active at very low concentrations and hence pose a high ecological risk, and also known to induce antibiotic resistance. And with the projected increase in reclaimed water usage and increased pharmaceutical production, this issue is compounded. Hence, addressing this will help us in addressing the sustainable development goals of food security, waste management, and sustainable agricultural practices. So, to move in this, with respect to in plants, these pharmaceuticals are known to disrupt plant homeostasis as stressors and also known to be food contaminants. So depending on the structure of these pharmaceuticals, they are actively or passively diffused in the roots and hence and travel through the plant via its xylem. And this uptake depends on the environmental factors, the physiological state of the plant, and also the soil properties. So to understand this mechanism, two attention has to be given. So we need to see what the drug does to the system and what the system does to the drug. So we have to understand both the pharmacokinetics and dynamics. To understand this, the important variables are there are pharmaceutical mixtures and there are not one or two pharmaceuticals. And these metabolites are even toxic than the parent pharmaceutical. And solely relying on experimental approach to see all these variations is challenging and little bit time consuming. And these fate of these molecules, to understand them, we usually use for analytical purpose 1,000 times more than the current natural concentration. So in terms of experimentation, we are only introducing these drugs again to the environment. So we need a new approach to understand the plant pharmacovigilance. So in terms of this, we see, as I said, there is numerous data on pharmacokinetics and dynamics, but comprehensively both has not yet studied. And also very limited focus is given in terms of metabolism and mechanistic insights of these pharmaceuticals in plants. And to address this, as our hypothesis is like these pharmaceuticals are like we are only design them, and we have rich toxicology data of these pharmaceuticals in animals. So why don't we leverage those data to understand the plant pharmacovigilance? Actually, based on a green liver model, and since professor also was discussing previously, plants and animals share a homologous anaerobic metabolism, and plants try to mostly detoxify these toxic compounds rather than mostly metabolizing them. And in terms of sequence metabolite patterns and enzyme classes, they are known to be homologous. And to use this, next we like now we are going to use green liver concept, and now to study the metabolism, genome scale metabolic modeling is an approach where a genome scale metabolic model represents a complete metabolic inventory of cell. And this genome scale metabolic models have been very successfully used for explaining abiotic stresses mechanism in other plants like Arabidopsis rice, maize, and even in microbial bioremediation, biostimulant strategies have been screened with genome scale metabolic models. And also with respect to human models, the effect of diet and liver toxicity have been studied with genome scale models, but this part has not been explored yet for plants. Hence, we thought of proposing a platform technology where we use green liver model and constraint-based modeling approach with genome scale metabolic modeling to understand the mechanistic insights. And to just give an overview of constraint-based modeling, so like plants are sessile. So they have to stay there and defend from the environment. So they have the metabolic plasticity, and they've evolved it to with robust solutions to meet its sessile lifestyle. So this robustness can be studied with constraint-based modeling. So like here we have a metabolic network which represents all the metabolic reactions happening inside a cell, and we connect it with reactions and all metabolites, and we write a mass balance in a and represent it as a stoichiometric matrix. And under steady state conditions with no accumulation, we solved it to get a flux space. So flux space is like for each reaction we have a range of values which this reaction can take. So comparing these two flux states between two different conditions will help us screen like what is the altered metabolism. So based on this, we give to reduce this flux space, we give constraints to the model, like biological constraints, like this is the maximum growth rate, like that this is the nutrients you should have. We will define the boundary, and we use an objective function, like say like the objective of the cell is to minimize the nutrient or usage, or objective of the cell is to maximize biomass production. Like that we give a biologically relevant optimization objective to target our flux space and compare between two different conditions. So we went ahead with this constraint based modeling. So to give and now to focus first as a model pharmaceutical, we went ahead with carbamazepine, and it is a anti-psychoactive drug which like used in neural disorders and all. And with this very low concentration, it is known to be teratogenic, and it's an emerging plant stressor. And as a plant representative, we used tomato plants, wherein for this carbamazepine we have rich data of its abiotic stress response in tomato, and being an agronomically important food crop, we thought of we'll see whether our model is able to match those predictions. And in tomato, CBZ is not only known to induce abiotic stress, but also are known to interact with insects, virus, whiteflies, how the plant interacts also. So since we have this data, we went ahead with this plant. And now to summarize it, so first we gave like a phototrophic input conditions where just a plant in a pot where it's able to exchange carbon dioxide, oxygen, and it has photons. And now we are forcing the plant to uptake the CBZ. CBZ is not used in its metabolism, so it's present in the environment. Now plant has no other way than to detoxify it. So we are forcing the CBZ uptake. And also with respect to the entire plant, CBZ with its neutral structure is known to be mostly transformed in leaves, and leaf is known to be like a significantly metabolically active part with respect to this carbamazepine. So first we went ahead with the leaf model. So to see that, to see that, like what we did is like first, as I said, like to make the model, first in the plants we know like the CBZ gets detoxified into these different metabolites. So we know A becomes B, but we don't know its metabolic reaction. So for that, we used data from green liver concepts from human pharmaceutical databases, and with respect to phase 2 and phase 3 metabolism, we went ahead with plant specific databases to write the glucose conjugation, glutathione conjugation, and its transport and deposition in vacuum. So, and then as I said, we were able to, like this model was even improved with the previous model, which was available, was improved in terms of gap filling and improved mass and charge balance to that stoichiometric matrix also was improved. And now, as I said, with the optimization, now the optimization given was the cell under these conditions should maximize the biomass and in minimal number of flux states. So that is for minimizing the sum of total flux. In minimal steps, it should maximize the biomass. So doing this, we were able to see with increasing CBZ, the model was able to predict decrease in biomass. And this was mainly due to the cofactor mediated competition. So endogenous metabolism and the plant detoxifying the CBZ was competing with the cofactors and redox carriers, inducing this decrease in biomass. And as I said previously, next, like now we have the model, and this model with leaf model is now capable of simulating the decrease in biomass. So next we went ahead with seeing with this decrease in biomass, can we predict the altered flux states? And after predicting it, like how can we apply to come up with the suitable biostimulant strategies? So, like now to see, with respect to CBZ stress, we compared two conditions, wherein control without CBZ and the CBZ flux inducing 50% reduction in biomass. We were able to see around 154 significant alterations in metabolic pathways, and of them, like we were able to hit upon the targets which are known to be present in tomato and which has known to be present in other plants, and also some new novel predictions we were able to make with the network level approach. And next to, thus we were able to induce this abiotic stress response. And then like plants are known to produce phytoprotectants like in terms of these stress, and these phytoprotectants when added as like exogenously, they are known to improve the CBZ stress. In terms of that, like we chose 4 biostimulants, purulin, glycerol, spermine and ethanol to see its effect in CBZ. We were able to see it is improving the CBZ stress with improved biomass in presence of them. Significant biomass precursors were enhanced in presence of these biostimulants, with proline being the highest. And we were able to see significant alterations in core metabolism, like improved nutrient uptake, photosynthesis, and antioxidant tolerance. Like it was improved in presence of these biostimulants, thus ameliorating the CBZ effect. And also we were able to simulate this crosstalk and come up with 144 significant alterations, and these 144 was like a part of those which were altered previously. And another interesting fact we observed is it is reducing the CBZ toxicity also, wherein with the presence of all these biostimulants, we were able to see improved phase 2 and phase 3 metabolites also. This was a very interesting observation. And thus like this is like a comprehensive representation of the entire model. So with this input, we were able to see the red highlighted ones are the critical bottlenecks, wherein if you provide them exogenously, like it will help in improving the CBZ stress. And this input is like carbon dioxide photon, it's coming, how this green highlighted box are individual biomass precursors. And thus we were able to comprehensively scrutinize the metabolic changes. And the same thing we extrapolated to see pharmaceutical mixture stress response also, wherein though the transfer metabolites not available in plants also, we were able to see its effect just based on these available data in human liver. And we were able to hit upon some targets of metabolism which are known in literature. And thus, to go forward, with the era of single transcriptomics available, more concerted efforts we can make to build more genome scale metabolic models of phytoremediating crops and apply this genome scale metabolic model to have a targeted experimentation approach. Thus, this will help us addressing and bring up rational strategies to maintain the like pharmaceutical, like agriculture yield amid the pharmaceutical stress. I thank my guides for the constant support and my lab mates of plant cell technology and computational system biology lab for their iterative improvement in my work, and my fellowship and family and friends. I thank you.
One short question.
Thank you, a very nice presentation. I have a very small question, like how long the CBZ will stay in the plants?
Okay, people have studied like till 80 days also. Mostly it enters via roots, and within mostly like 2 days it goes till leaves. So there are different parts, like it's a part of it gets transformed, part of it gets accumulated, and it stays forever. And the CBZ has an half-life of around 120 days. Both simultaneously happens, ma'am. It sometimes accumulates also and get transformed also. That percentage, I don't think we can. It depends on different factors.
Okay, fine.
Yeah.
All right, thanks.
The next one.
Yeah, thank you, Samyukta. Thank you for
Is not there. We have the
Very nice
PPT,
work.
so we put the slide rapidly. You watch, we move, and we move to the. No, we move. No, there is no. No, we just pass.
Yeah,
but we cannot evaluate. We cannot evaluate.
No, we'll have. Yeah, we have a presentation from PM Varuni, but she did not send. She is not available, and she did not send the presentation. Only her PPT is available. So we'll just play the PPT, and we'll go to the next presentation. Just 2 minutes. But yeah, actually she has sent the presentation. So when she 1 minute, maybe within 1 minute we'll see, so that we'll have some idea what she has done. Okay. Yeah. It took only 1 minute.
Ah, it's also a record session. Ah, it's also record. So just listen.
It's only recording.
Hello everyone, I'm Prama and I'm a PhD candidate in environmental studies at Queen's University. My supervisors are Dr. Barbara Zeeb and Alison Rudder from the Royal Military College of Canada and Queen's University respectively. So petroleum hydrocarbons, or PHCs, are straight-chain or cyclic organic pollutants found in oil and petroleum products. Within PHCs are often polycyclic aromatic hydrocarbons, PAHs, comprised of 2 or more fused benzene rings. And larger PAHs are of concern as they may be carcinogenic or mutagenic at high concentrations. The goal of my research is to create a framework for the phytoremediation of PHC contamination in the Canadian boreal ecozone using native plants, symbiotic mycorrhizal fungi, and rhizosphere-dwelling bacteria called rhizobacteria that participate in nutrient exchange with the plants. I'm interested to see if the presence of the symbiotic mycorrhizal fungi and rhizobacteria enhance the phytoremediation process. So I collected coarse-grained background and contaminated soil from the site of a former Canadian petroleum terminal in 2022. The site is located in the Canadian boreal ecozone and has extremely high levels of PHCs from oil spills at a concentration of approximately 26,000 milligrams per kilogram, despite not being active since the year 1998. This means that natural weathering processes were not enough to remediate the site over the last 25 years. And in this table here, I break down the total petroleum hydrocarbons in the soil by fractions and compare against federal and provincial guidelines. So fraction 1 contains the most volatile PHCs, while fraction 4 contains the largest and most recalcitrant ones. As you can see in the red rows, the background soil from the site is clean, whereas the contaminated soil has significantly higher fraction 2, 3, and 4 levels than both federal and provincial guidelines, as highlighted in yellow. And this is an environmental concern for us and is also why I am interested in developing a strategy to remediate the site using greenhouse trials that will eventually be applied to the field site. I am working with 5 native plant species. Picea mariana is a tree, Achilla millefolium is a forb, and 3 tufted grass species, Andropogon gerardi, Bouteloua curtipendula, and Panicum virgatum. All species have shown tolerance to PHCs in past studies. And I'm using 2 soil inoculants. The first, a commercial blend of arbuscular mycorrhizal fungi of the Rhizophagus genus. So these fungi form symbiosis with approximately 85% of all terrestrial plants, including my 5 species. And I've also cultured the non-pathogenic and plant growth promoting bacteria, Bacillus subtilis. And the strain I'm using is non-pathogenic, highly resistant because it forms endospores, and is especially known for producing lots of biosurfactants. It resides in the soil and may be also taken up by the plant as an endophyte. Biosurfactants reduce the surface tension of petroleum in water, facilitating the microbial remediation process by increasing soil water retention and PHC bioavailability to PHC metabolizing microbes. In January of 2023, I started the first half of my 2-year greenhouse experiments. I planted seeds of the 5 selected plant species in the background and contaminated site soil in the greenhouse in 2 separate treatments. The first was a plant-only control, where I just grew the plants from seed in the soils. And treatment 2, I inoculated the soils with AMF and then grew the plants. And due to the very high concentration of PHCs in the contaminated site soil, at nearly 26,000 milligrams per kilogram, I created a 1-to-1 mixture of background and PHC contaminated soil, which I refer to as the diluted soil. And this concentration was about 13,000 milligrams per kilogram total petroleum hydrocarbons, half the total for the contaminated soil. And I used 2 of the best growing grass species, Andropogon gerardi and Bouteloua curtipendula. And then I subjected them to 4 treatments in the diluted and background soil starting in June of 2023. This includes the first 2 treatments, and also for treatment 3, I inoculated the soils with freshly cultured Bacillus subtilis, and for treatment 4, I inoculated the soils with both the fungi and the B. subtilis. And I wanted to make sure that the B. subtilis I cultured was healthy, active, and very high in volume. So to do so, I monitored their growth using absorbance and cell dry weight readings, which I used to create growth curves within the linear range. And I created 2 growth curves by culturing a seed inoculum using nutrient broth, and then inoculating 6% of the culture from the seed inoculum into a custom-made mineral salt medium. And mineral salt medium was used because it's known for promoting biosurfactant production in B. subtilis. I used sucrose as the carbon source for the mineral salt medium because it's a common plant root exudate and it's also found in soils. So as you can see in the graphs, like most bacteria, B. subtilis first experiences a log phase where it adapts to the environment and is not growing very much. Then it experiences an exponential phase of growth and then finally reaches a stationary phase of growth where the growth essentially plateaus. And I inoculated the treatment 3 and 4 soils using B. subtilis in the beginning of the stationary phase of growth from the mineral salt medium culture, where their numbers are very high and they're still very active rather than falling asleep in the culture. And then I determined the critical micelle concentration of the mineral salt medium culture to determine how efficiently the culture can produce biosurfactants to facilitate the phytoremediation process. And I observed the critical micelle concentration at approximately 1.6% volume per volume culture in sterile water. As micelles were formed at such a low culture volume in water, this indicates highly efficient biosurfactant production by the B. subtilis culture. And I'm periodically taking measurements of plant height throughout the course of my 2-year experiments as an indicator of plant health. So these graphs show the mean plant height, or above ground length, of the plants in the treatment 1 and 2 pots from the 5 plant species, which I started in January of 2023, using the background and contaminated soil. And the plants here have been growing in the soil for approximately 1 year. The light and dark green columns represent growth in the background and contaminated soils, respectively. I conducted Mann-Whitley U tests to compare above ground height in the background and contaminated soil. As you can see, 4 of the species, except for the slow-growing spruce, Picea mariana, are growing very well in both treatments. The fungi improved growth in the contaminated soil for Andropogon girardi and Panicum virgatum. Here is a progression of the same plant another 6 months later. Here, Butylua curtipendula and Panicum virgatum are growing significantly taller in the background soil in treatment 2, but that's not the case in the contaminated soil, which is causing the significant growth difference in treatment 2 soils for these 2 species. And here I show the 2 grass species, Andropogon girardi and Butylua curtipendula, growing in the diluted soil after 10 months of growth, which occurs at 1 month after inoculating the soils with B. subtilis. This means the plants were growing for 9 months before I inoculated the soils with the bacteria. There's no significant difference in Butylua curtipendula height in all 4 treatments, which shows that the species can tolerate the diluted soil very well. Here are the same plants 3 months later. The only difference in growth between the background and diluted soil is that Andropogon girardi in treatment 4, where we see the species growing significantly taller actually in the diluted soil rather than the background soil. And it's interesting to see at this point that the treatment 4 plants are not growing taller than the control plants in treatment 1, despite being inoculated with 2 plant growth promoting symbionts, being the AMF and the B. subtilis. So I suspect there may be some competition between the plants AMF and B. subtilis for soil nutrients, which may limit plant growth. Alternatively, perhaps the pH levels in the soil are impacting plant growth more than the soil inoculants used. So I'm in the process of determining the current pH levels in the soil to confirm or deny this. This summer, I also extracted bacterial DNA from the rhizosphere soil samples in order to identify soil bacterial composition in my greenhouse pots. I'm interested to know how the composition differs among the 4 treatments and which treatment shows the most bacterial biodiversity. I followed a standard 16S metagenomics approach because the 16S rRNA gene is ubiquitously found in bacteria. First, I extracted the total DNA from the rhizosphere soil samples using a renowned commercial kit, and I then verified the success of DNA extraction using qubit fluorometry. Following this, I amplified the 16S rRNA gene using qPCR and enumerating the started the starting bacterial DNA quantity using a standard curve and critical thresholds. I then denatured and diluted the purified double-stranded bacterial DNA so that I could perform next-generation sequencing on the aluminum MiSeq. So currently, my sequencing results are being demultiplexed using bioinformatics so that I can identify the different bacterial genera in each treatment. And in the meantime, I can compare bacterial DNA quantities from the different treatments by analyzing results from the qubit and qPCR analysis. So treatments 1 and 2, which were the control and the AMF treatment, had the highest total DNA extracted. And generally, the contaminated and diluted soils contained more bacterial DNA than the background soil, which leads me to believe that perhaps hydrocarbon metabolizing bacteria are highly prevalent in those soils. Among the treatments, treatment 3 and 2 had the highest quantity of bacterial DNA in both the background and diluted soils. So treatment 3 was directly inoculated using only Bacillus subtilis. And for treatment 2, I inoculated the soils with AMF, which does support the growth of many rhizobacteria. But it was surprising to me that treatment 4, where I inoculated the soils using both B. subtilis and AMF, had the least amount of bacterial DNA. And I think this may be because B. subtilis and AMF compete for several of the same soil nutrients. So it's possible the AMF outcompeted B. subtilis for soil nutrients, or that B. subtilis was taken up by the plant into its tissues as an endophyte. So in summary, the addition of AMF significantly improved plant growth a year into growth. And the plants are also growing more in the diluted soil compared to the contaminated site soil, which makes sense because the pH levels are lower. Around 18 months into my greenhouse experiments, I start to see no significant difference in plant height between the background, diluted, and contaminated soils and the 4 different treatments. This may be due to plant tolerance to PHCs after the first year of growth, during which time bioremediation likely occurred and PHCs could have been detoxified, which I'm in the process of determining by measuring the PHC levels in the soil. And finally, my qPCR data revealed that treatment 3 has the most rhizosphere bacteria, which I attribute to inoculation of soils with B. subtilis. Treatment 4 has the least rhizosphere bacterial DNA, which may be due to nutrient competition between B. subtilis and the AMF. So next year, at the end of my 2-year greenhouse project, I will harvest all of my plants and measure their above ground and below ground biomass, and this will be an informative metric on the impact of soil and the treatment on plant health. And I'm also measuring the final PHC levels in the soil to determine which treatment was most successful in remediating the PHCs. Finally, the best growing plant species in treatment for phytoremediation will be used together in a pilot scale plot at the site to determine how effectively they can remediate the soil in situ. And if the pilot scale plot is successful in remediating the PHCs, the selected plant species in treatment may be used together in commercial and industrial purposes to remediate other PHC contaminated sites across the Canadian boreal ecozone. And ultimately, the final intended outcome of this study is healthy plant communities and soil ecosystems following phytoremediation. So thank you all for your time.
I think this is the end to the session. So we'll all quickly go to the poster sessions now, and then we'll start the poster session. Thank you all. Yes, everyone should be there.